I'm just starting the live stream, yeah? You should see the live on YouTube option.
Hi, Poonam.
Hi.
Want to get started, Anupama?
Yeah. Shall I go?
Yep.
Okay. Okay, so hello everyone. Welcome to the second day of the NeuroFemIndia 2021 conference organized by Bias Watch India.
Oops,
This
did
conference
we end up missing?
Hey, Shruti, is something happened?
No, you should carry on. Sorry, I think there was some feedback.
Oh, okay, sorry. Okay, so yeah, hello everyone and welcome to the NeuroFemIndia 2021 conference. This conference is being run in collaboration with the ALBA network, which has generously agreed to sponsor the event, and our media partner InsightCom. So I'm Anupama Satyamurthy. I'm a DBT Ramalingaswamy Fellow at the Center for Neuroscience at the Indian Institute of Science, Bangalore. I'll be moderating today's sessions with Dr. Shalagna Das, who is an assistant professor at the Albert Einstein College of Medicine, New York. So both of us are members of the core organizing committee for this conference. And volunteers Arushi Bhatta, who's a PhD student at CSIR IIIT New Delhi, and Annapurna, who is a PhD student at CSIR CCMB Hyderabad, will be helping us with the technical requirements of Zoom and ensuring that we can have a productive interaction with our speakers and all of you, our audience. So I just wanted to quickly go over the events for the day. As you would have seen in our event posters for the day, we're going to have public talks by 6 speakers, followed by private breakout rooms from 4 to 4:30 p.m. IST for extended discussions between the speakers and selected members of the audience. So since we wanted to maintain a small number of participants to enable meaningful breakout room interactions, we prioritized trainees and students to participate in these. But in the spirit of building community, we encourage all participants to reach out to the speakers for any additional questions. After that, we're going to host the ALBA network session on bias in India STEM. So this is going to be a full and interesting day. So let's get right into it. Right before that, we have 2 general notices for the audience. At this point, we would like to remind you that we are also live streaming this conference on YouTube. On Bias Watch India's channel for any members of the public and the Indian academia who have missed out on registering for the event. The link should be displayed in the chat. We'll not be accepting any audience questions for the field overview talk, but for all the other talks and sessions, we highly encourage the audience to ask their questions in the Q&A pop-up box. So please add your questions to the Q&A pop-up box rather than the chat box so that we can find them easily. And just 2 more things here. Please upvote the questions that you want the speakers and the panelists to answer. And please address your questions to specific panelists by name. Okay, so starting off the talk series today is Dr. Shashi Bala Singh. So please join me in welcoming her to the stage. She is currently the director of the National Institute of Pharmaceutical Education and Research, Hyderabad. And prior to this was the Director General at the Defense Research and Development Organization, New Delhi. So apart from her considerable accomplishments as a research administrator and journal editor, her interests have focused on neural correlates of high-altitude human physiology and oxidative stress-linked degeneration, both in terms of fundamental and translational research. She is the recipient of many awards and felicitations from the Indian government and is also a member of the DRDO Inter-Academy Panel on Women in Science in India Vision Document. So today she is representing the field of systems and computational neuroscience, and will give a talk focused on the history and importance of this field. So Professor Shashi, thank you for accepting our invitation to this conference. The floor is all yours.
Yeah, thank you for a nice introduction, and very good afternoon to everyone, especially the panelists and to the participants who are joining us virtually. And I also want to thank NeuroFemIndia 2021 for organizing a women-centric conference on women in neuroscience of India in particular, and maybe also some people have also joined virtually from other part of the globe. Today, the point of discussion is computational neuroscience and system neuroscience. You know, when we look at the brain, I always wonder this marvel which has been created, you know, which is just 2% of the body weight, but wants 20% of the oxygen consumption of the body. Still, it's an enigma to know what brain is all about. How does it function? How does it sort of integrate different kind of activities in different parts of the brain and bring out some kind of effective behavior? I always feel brain is a chemical computer. And it is the software of the body, and the body structures is the hardware of this. And it is a chemical computer which defies all the body actions which we see in an individual in a very integrated and coordinated way. If there is some problem, it is because of the imbalance of the chemicals or the neurotransmitters which are released in the brain. And because of this imbalance, we can have different kind of problems at system level, at organ level, at cell level, and maybe at other levels also. So it's a very complex organ of the body, and to understand it is a very big challenge. But then every challenge has a lot of opportunities. I think this is a good time for all the women on working in neuroscience to be part of this challenging journey, so that we are able to explore, if not everything, some part of it. And if you see, you know, when we have to understand brain, it is almost all the sciences converge together to make us explore, to understand how the brain functions. Anatomy, physiology, physics, electronics, computational biologists, biochemists, biophysicists, mathematicians, and many more sciences have to be part of this journey to understand how does brain function. Research in neuroscience is what we have to talk, and the history of neuroscience, the way it has grown in India. I think if you look at the neuroscience in India, I think we are making good progress. But then there is always a lot of room where we need to work harder and make it to the top. And when we talk research in neuroscience, you know, the computational neuroscience and system neuroscience are the two main components of neuroscience. Computational neuroscience is mainly a theoretical approach to understand how brain processes information, while system biology or the system neuroscience is a science where we need to integrate all the processes together and look at the effective behavior, which can be in terms of many functions which can be corroborated with the effective behavior. And if you see this term, system biology, it originated in the mid-90s, but you know, as we are evolving, this system biology or system neuroscience is getting a lot of importance, because I can tell you whatever we do in science, whatever we are able to explore, whether at the gene level, molecule level, or at the organ level, finally we have to test it in the body system. Finally, it has to be integrated, coordinated approach to understand how the intervention helps the body system in bringing out the effective or the desirable behavior. And with time, the way things are getting, like genomics, genotyping, then proteomics, all type of omics, they have led to a paradigm shift in understanding system biology. So to my mind, this system biology, omics, they are the very powerful tool to understand how all systems work together to give an effective behavior or the outcome in an individual. Neuroscience in India, as I said, started almost in 1970s. And I think NIMHANS, National Institute of Mental Health and Neuroscience, which predominantly came into existence in terms of working, terms of doing research in neuroscience. They initially focused on the clinical focus, and it was established in 1974. Thereafter, for the first time, CDRI Lucknow conducted International Symposium on Central Scientific Transmission. And this was the probably first symposium conducted in India on the neuroscience by Indian scientists. And it almost took 2 decades or more for the formation of Indian Academy of Neuroscience, the primary association of neuroresearchers in India. And this Indian Academy of Neuroscience was established in 1991 with a lot of stalwart coming together like P.N. Tandon, P.K. Seth, Mehdi Hassan, Professor Dhawan. And now if you see the influence of Indian Academy of Neuroscience, it has grown not only in size, but also in the influence which it has on the young neuroscientists, not only working in India, but working abroad. Every year when we conduct this conference on neuroscience, we have large number of participants, large number of presenters who become part of this activity. And it's a good mela, celebrations which we have, and we want to understand and also see what are the challenges and how far we have come in terms of understanding neuroscience in different parts of the world. Also in 1951, we also had Neurological Society of India. It was mainly formed by the clinicians. However, it is also there and they are also doing a good job. But I've seen in the last 2 decades, neuroscience, Indian Academy of Neuroscience is doing very well in terms of number of participants, in terms of it has influence, not only in the Indian continent, but globally. And how the science has, or the neuroscience has grown, initially, as I said, NIMHANS was the first to take up research in clinical neuroscience. Then we also have many more institutes with joint hand, like NCBS, Indian Institute of Science, lately IIT Madras, IIT Mandi, IIT Ropar. If you see, some of them have good center in computational neuroscience. Then PGI Chandigarh also developed a low-cost, low magnetic field MRI for point care of testing and associated CAD system. Similarly, there are others also, like Jawaharlal Nehru Center for Advanced Scientific Research. They, with the help of NIMHANS and CMAP, Center for Cellular and Molecular Platform, have identified a molecule that could clear toxic aggregates in the brain and reduce the loss of neuron. So there's a lot has been done. I will not be able to talk about everything, but I'm sure people are working. But we need to have much more concentrated effort to take things forward in the area of computational neuroscience and also in the area of system neuroscience. And we not only there are neuroscientists collaborating within India, but also we have some of the neuroscientists are collaborating with the national partners, and they are doing a good job in understanding many aspects of brain. How does brain function, especially memory, attention, perception, cognition, awareness, thoughts, language and consciousness, and many other things which we need to understand, because as I said, it's still is an enigma. How does brain function? If we know how does brain function, I think we can always build on the pathophysiological changes, and that can be a way to have the therapeutics to sort of mitigate those changes. One thing which I want to emphasize, especially to my young neuroscientists, it is a time to do a quality research. I think quantity, if you look up 2 years ago, we are 9th in number of paper we are publishing in neuroscience. But I think it is just not the number. We should be seeing that how does we improve the quality of science which we are doing. For this, it's very important that our young neuroscientists should have mentors and mentees which are doing much, much better in this field. And this is the way we'll be able to improve our quality of research in the neuroscience. We have to move from volume to value. Other thing which I've been asked to talk about, the computational neuroscience. It is again an emerging area, very important area. However, we still have to pick it up in Indian context. There are many centers when I was going through the internet where this computational neuroscience is being done. And this computational neuroscience started almost in the second half of 1980s. And the term was coined by Schwartz, Eric L. Schwartz. And as we know that since we came into computational neuroscience only very late, but then if you have heard of Huxley and Hodgson model and how does nerve potential, works and the mathematical modeling and computational neuroscience, this is how this computational neuroscience started with this model. And now we have many things which has been done, like introduction of integrate and fire neuron values, which has been there as a part of computational neuroscience. And next step was the work of Wilfred Roll, who used mathematical approaches based on cable theory to show that dendritic arborization neuron strongly affects processing of synaptic input. In India, we have, as I said, there are a lot of centers, like I could see some of them. IISER IIT Jodhpur has a good computational neuroscience center, NBRC, AIIMS Delhi, IIT Madras, Indian Institute of Science. And some of them are doing a good job. But then I think we need to focus on this because for the reasons we are entering the age where we have large volume of data. If you see the sequencing genome, like if you do mRNA sequence, gene sequencing, and also omics, which has come like metabolomics and all this. So we are going to generate large number of data. So there are 3-4 challenges which we are facing as a computational neuroscientist also, for which we need to integrate data mining, artificial intelligence, machine learning, brain-computer interface. Only then we'll be able to take this science forward, and there's a lot of scope because nowadays when there's a lot of volume of data is there, how do we sort of integrate it, process it to understand how it will help in terms of prediction of the prediction of a disease, preemptive, and also prognosis of a disease. All this through the computational neuroscience only will be able to sort of understand the predictive, preemptive, and therapeutic or prognosis approach of treating different diseases, not only in disease pertaining to the brain, but disease pertaining even to the system biology. So at present, I strongly feel that in our present ecosystem, the country, it favors basic research in academia, while translation research is confined to clinical science and industry. If you see, most of us are basic researcher, but being in DIDO, we had the focus was to bring out product and processes which can improve quality of life of the soldiers which are posted in different stressful environmental conditions. So we could bring out some product which has sort of try to make them in a slightly comfortable ambience in those stressful environmental conditions. But since academia forms a large bulk of researchers, there is a need to create appropriate ecosystem to foster translation research in academia. Important to have right balance between basic and applied research for larger benefit of the mankind. See, whatever we research do, I think all of us should have one thing in mind, that it has to finally translate and go to the common people, go to the masses. They understand what we are doing, not only in terms of the science as such, but also how will this science help a common man to improve his quality of life. We need to bring all discipline together. As I said, neuroscience, it is multidisciplinary, multi-institutional research has to be carried out. And I think things are moving in this direction. And now I see many projects coming from many institutes with different kind of expertise, and that really help people to do a good science. So as I said, all this computational neuroscience, we need to have artificial learning, brain machine artificial intelligence, machine learning, which will help us understand the neuroscience, also help us treat people with different kind of challenges which they may have. Second challenge is the translation neuroscience. What is happening? We are working like a 6 blind men. They are watching they are feeling how is the elephant. So out of 6, one touches the legs and feels a tree trunk. And other one who is holding the tail, he feels it's a snake, like a snake. So we have islands of excellence. I think we need to move towards continent of excellence by bringing a holistic approach to the neuroscience and to all the sciences in general. So it is a time to see elephant as an elephant. Need to synergize between all branches of science to address the problem statement. And we should be working for a big tent model, not only the basic neuroscientists, applied, translational, clinician, and policymaker should all be working together in a synergized function to address a statement problem and to also come out with some kind of solution. With this, I want to thank NeuroFemIndia for including me in your program. I look forward listening to other talks. I think I also have to introduce my next speaker, Dr. Pinky Kain. Am I right?
Yes.
Yes. So I have a lot of pleasure in introducing Dr. Pinky. I was going through her biodata. She is very young, yet she has made a remarkable achievements in her career. Dr. Pinky is just excuse me. Pinky, she is now a fellow of the Wellcome Trust DBT India Alliance at Regional Center for Biotechnology, NCR Biotech Science in Faridabad. She has obtained her PhD from NCBS for Bangalore and has been on postdoc to many countries like Germany. Then in a year, she has worked with the University of California, and also she is visiting scientist at Buck Institute in California for 2018 and 19. She has a lot of awards to her honor. She has traveled widely to attend conferences and present her paper, like Travel Award for ESITO, then IBRO Travel Award, and full fellowship for 8 Japanese Drosophila Research Conference. She's also the editor of Acta Scientific Neurology and Associate Director of Science Progress and Neuroscience Insights. She has been awarded, she also has a patent to her credit at this young age, and this really speaks that how she's engrossed in trying to translate her research finding into some kind of product. I wish her best, and I hope in coming year we will be hearing that some product has been launched by Dr. Pinky. Dr. Pinky, she will be delivering a talk on gustatory processing, understanding taste and its modulation during Drosophila as a model system. Over to you, Dr. Pinky.
Thank you, Dr. Shashi, for a very kind introduction. So it doesn't allow me to share the screen. I'm trying to share my screen, but I can't do it.
Vaishnavi, could...
Yeah, can you check?
Yes, now it shows. Thank you.
Thank you.
Okay, so good afternoon, everyone. And before starting my talk, first I would like to thank the organizers of NeuroFemIndia for such a wonderful initiative and opportunity to celebrate Indian women in neuroscience. I'm Dr. Pinky Kain Sharma, and I'm currently working at Regional Center for Biotechnology, Faridabad. My lab is interested in understanding the peripheral and central coding, taste coding, and how the taste information is wired and processed in the brain, and how it gets modulated by various intrinsic and extrinsic factors. So as we all know that taste is extremely important for all the organisms to evaluate and choose foods that are rich in calories and avoid bitter compounds that could be toxic, taste or feeding behaviors are regulated by our appetite, and events that takes place at the periphery in the body and in the brain, they control our appetite. So the synchronization of events at the periphery and in the brain are actually very important for the proper regulation of our appetite. And when the appetite is disturbed, these events become desynchronized and cause metabolic conditions like obesity, diabetes, cardiovascular diseases, and hypertension. So in order to understand how the taste information is perceived and processed, what are the central taste pathways involved, and how the internal state helps in giving rise to simple behaviors like acceptance or rejection of food, we are using Drosophila as a model system of our choice, which you can see here, they're playing in a small arena. So we are using flies to understand feeding behaviors because both human and flies, they share similar taste organization logic by which taste behavior and food choices made. Although fly gustatory receptors, they share no homology to mammalian taste receptors, but both share similar preferences to foods, and both are capable of differentiating basic taste modalities like sweet, bitter, sour, salt, and umami. So in humans, we know that the tongue is the main peripheral taste organ that holds the taste receptors. Taste receptors are housed in the taste buds, which you can see here, and they are present on the surface of the tongue. So the incoming food substances, they bind to taste receptors present on the surface. They activate the cell's internal working network or pathways that sends a signal to the brain about palatability, flavor, or editability. And multiple cranial nerves transmit this information from the mouth and the pharynx to the brain via the brainstem. So if we look at the invertebrate model systems like flies have a similar taste organization logic. Flies detect different taste compounds in their environment by using various peripheral taste organs, including labellum, tarsus, genitals, and wing margins. They also have pharynx, which have three different internal organs called LSO, labral sense organ, ventral sebareal sense organ, and dorsal sebareal sense organ. And pharynx have hairless sensilla. So since taste is a form of chemoreception, the gustatory here in flies, they have a pore to allow the taste solution make contact with the taste neurons present inside, and each sensilla does generally have 4 taste neurons and 1 mechanosensory neuron present inside. The role of taste specs and pharynx is still underexplored, and the distribution and morphology of taste sensilla present on the labellum is remarkably stereotypical between individual flies. So from periphery, which you can see here in this confocal image, all the taste neurons, they terminate inside the brain in a special area called subesophageal zone, or SEZ, and they basically terminate in very non-overlapping areas and show discrete patterns. So just by looking at the expression pattern, you can say, oh, this neuron is coming from leg or pharynx or the labellum. And SEZ is the first relay of taste information in the brain. So we know that food preferences involve the peripheral taste system, but decision-making occurs inside the central nervous system, and various physiological signals and other sensory information act on these circuits which are present inside the brain or the peripheral neuron. So in contrast to the extensive knowledge on the peripheral coding of taste, both in flies and mammals, much less is known about the central pathways and neural circuits that integrate and translate these signals into meaningful behavior. How specific taste neuronal circuits in the brain influence feeding behaviors is still not clearly understood, especially the ones which are involved in forming taste memories. So unraveling taste circuits, therefore, will be important not only for understanding how sensory input is translated to behavioral output, but also how taste associations are formed in reward and aversive learning, and to understand the neural basis of taste preferences. My work previously has identified a novel class of second-order projection neuron, which you can see here in green, cluster 1. And these neurons, they make synaptic connections with the peripheral GR5A positive sweet neurons. The cell body of these projection neurons, they are present around antennal mechanosensory and motor centers, also called AMMC. As shown in this 3D model, the neurites of these neurons, they terminate in a very characteristic pattern, just like sweet gustatory neurons, which come from the periphery here. And what our calcium imaging measurements suggested that these sweet projection neurons, they have a very dedicated role in processing only the sweet taste information, nothing else. We observed a very robust concentration dependent increase in calcium activity, which you can see here in the graph from the MMC area, not from SEZ. So this suggests that perhaps the second order projection neurons relay taste information from the SEZ to MMC. And since MMC is MMC receives input from mechanosensory and other olfactory neuron, it appears that MMC is actually involved in processing multisensory information. What we also found is that the activity of these projection neurons modulate by starvation and dopamine, and all together, our results indicated that the activity of these neurons, they get modulated by hunger in a manner that can communicate altered reward value to higher brain centers. So to understand how specific neural circuits influence feeding behaviors, we found many putative central neurons in the SEZ in a screen done in the lab, which you can see here. And most of them make synaptic connections with the peripheral sweet neurons, and also the inducible activation of these neurons by just expressing heat sensitive cation channel trip A leads to extension of proboscis in fly at high temperature. And all these neurons are just present in the SEZ, nowhere else. We have also found some novel circuits for bitter taste, which are not shown here. So far, only a handful of central taste neurons have been found in flies, and now we are working on the circuits or the microcircuits that we have found in our lab and trying to understand their physiological role in fly. So apart from what I just showed you for sweet projection neuron, there are various factors that can act on taste circuits and taste cells and modulate the feeding choices and behavior, just like thirst, satiety, dietary restriction, use of drugs, metabolic disorders, prolonged illnesses, aging, or neuropeptides and neurotransmitters. So one such factor that we are studying in the lab is table salt or sodium chloride, NaCl, which is an essential component of our everyday diets. Just like sweet substances, animals have an innate liking for salt as well. And salt is considered mineral taste, as stimulus is just a simple ion. We know that sodium is an important cation, and our body strictly regulates and maintains sodium levels in the body to support important functions like nerve transmission, prevention of muscle cramps, absorption of nutrients, putting muscles into actions. And animals, since cannot metabolically create sodium themselves, ingestion of this ion is important to be taken through external food sources to carry out these various physiological functions. And a specific appetite for salt has been shown in other animals too, not just in humans, just like this, what you can see here, puddling of butterflies in urine patches, cows licking rock salts, and Mormon crickets cannibalizing each other, mosquitoes sucking human blood to get the sodium. Even during the periods of reproduction in many animals, including humans, an increased need for both sodium and protein has been seen. Eating high salt is a serious concern, and it leads to a number of diseases in humans. And despite of this burden on the society that I'm showing you here, we currently lack enough knowledge about the neuronal pathways, taste organ, receptors, circuits, and genes that regulate high salt consumption. We still do not know how high and low salt perceptions are differentially encoded. And recently it has been shown in mice that mice use bitter and sour pathway to detect the high salt concentration. So the one big question that arises is that if high salt is a part of aversion pathway, why do humans even like high salt? So to understand how dietary pre-exposure to high salt changes or alters subsequent feeding behaviors, we started looking into the mechanisms that regulate high salt feeding in Drosophila, which has not been explored so far. So first, what we asked is how wild-type flies respond to different concentrations of salt. And for this, we used a two-choice feeding assay, which you can see here. Flies, we generally allow the fly to eat red or blue spot. Red is with salt, and the blue is just plain agar with water. And then we look at the fly's abdomen to ask what exactly flies were eating. So in these assays, what we found that wild type flies are attracted to low concentrations of salt, generally 50 millimolar. And with the increase in salt concentration, the salt becomes aversive to flies, suggesting attraction and repulsion to salt is a concentration dependent phenomena. The similar feeding responses were observed when the mated male and mated females were tested separately, and also in our spectrophotometry assays. So in general, what we found that feeding responses in mated females were higher compared to males, suggesting mating induces a salt appetite in Drosophila females by increasing the gustatory response to sodium. Next, we did an independent assay called proboscis extension reflex assay, or PER, to confirm our results. Extension of proboscis is a first step in the appetitive behavior. In this assay, we provided a series of NaCl concentration to the tarsi of the fly or to the labellum taste hairs. So these were the similar concentrations that we used in our feeding assays. And remember that the taste neurons are also present in the fly leg. As shown in this video, after stimulation of leg taste hair of flies with low concentration, flies show an extension of the proboscis. We found that highest PER responses again at 50 millimolar salt, and as the concentration increased, the responses were going down. And in all of these assays, we did not allow the fly to drink the solution while presenting the stimulus. So our PER results again suggested that low concentrations are attractive and high salt concentrations invoke aversive behaviors in fly. To understand the effect of dietary pre-exposure to high salt on feeding behavior, we designed an approach where flies were fed on normal media and treated as control fly. For test flies, these flies were fed on high NaCl diet, which is 200 millimolar salt in normal media. And after 3 days of feeding, following starvation of 24 hours, these flies were tested again for various concentrations of salt. So what we found that generally 50 millimolar is an attractive concentration, and then the responses decrease with increase in concentration. In flies which were feeding on high NaCl diet, the responses were shifted towards the high salt concentrations. And this reduction in the responses was enhanced. So the aversion was enhanced, and flies all of a sudden started liking high salt concentration. So what this result suggests that maybe it is the internal state, because we are using the star flies that are responsible for modulating this kind of salt behavior. So next we asked if it is indeed the internal state, what happens in flies when they were not starved. So when we use the fed flies, surprisingly, we did not see an increase in high NaCl fed flies, and in general in both the conditions the responses were quite low, suggesting that internal state indeed guides the flies feeding decisions. So our results also highlights that a single taste stimuli, which is a salt here, can drive opposing behaviors by activating distinct taste pathways modulated by internal state, like hunger in our condition, and shifts the behavioral response from aversion to attraction, as seen here, possibly by enhancing the appetitive pathways and suppressing the aversive pathways. We also found increase in total mean weight of mated females compared to mated males after feeding them on various concentrations of salt, suggesting perhaps in mated females and males, salt is perceived and metabolized differently, and also mated females develop a very strong specific appetite for sodium following mating to support the egg development and to invest in the progeny. There is also a possibility that high salt feeding leads to obesity in mated females by making female flies resistant to neuropeptide F that signals brain about satiety. We wondered next what high salt do to other taste preferences, and for this we tested sugars, bitters, and many other salts. So we did not really see any changes in bitter and other categories of salt, but in case of sugars, what we found that flies started showing enhanced feeding preferences for very selective sugars, like low concentration of sucroses and 100 millimolar concentration of fructose and glucose, which all these sugars are easily metabolized by flies. And it is interesting to observe that the feeding on high NaCl is actually altering and modulating behavioral sensitivity towards the sugar, maybe to balance the nutrient requirement in flies, or maybe something else which we don't really fully understand yet. So from the peripheral system, what we know that flies have 68 taste receptors. More than 50% of these GRs are predicted to be bitter receptors, and we really don't know the ligand for many of them. What is shown here is a sugar clade which has 8 members, 8 sugar GRs that identifies various type of sugars. GR5A and 64A shown here, they are necessary for almost all sugar detection. And apart from these GRs which are shown here, there are inotropic receptors, pickpocket genes, TRP and ENAC channels which have been shown to detect various other categories of taste. So to understand why do we see enhanced feeding responses and modulation of taste behavior when flies were pre-exposed to high salt conditions in diet, for this we started looking into different categories of taste neurons. Just to remind you again, what I had shown in the beginning, that there are 3 distinct internal taste organs which are present in fly pharynx, namely LSO, VCSO, and DCSO. After the food intake is initiated, pharynx controls the ingestion of food and encourages only intake of appetitive food in flies. We know that neuronal activity in neurons play a key role in the development and maintenance of neural circuits. It also controls synaptic stability and the rate of synapse formation, and regulates the localization and trafficking of synaptic vesicles and neurotransmitter receptors. So what we did first was to look at the feeding behaviors in the absence of neuronal activity by shutting down the neuronal activity first in the sweet neurons. And to do this, we use active form of the tetanus toxin. We express tetanus toxin in the peripheral neurons of the fly labellum. So what you can see here is GR5A marked sweet neurons which are present in the labellum but not in the pharynx or anywhere else in body. So when we tested the responses of these flies where the neuronal activity was knocked down, what we found, these flies, they showed high preferences for low and high concentration of salt compared to their parental controls, which behave just like wild type flies. So interestingly, what we observe is that blocking the neuronal activity at the periphery in these neurons leads to high preferences for low and high salt in flies, which were pre-exposed to high salt diet. Next, we tested many other gustatory neurons that express not just in labellum, but also in the pharynx or also in certain combinations in pharynx and at the periphery. So one common features in all these receptor neurons is that all of them express in LSO. Each one of them has some expression in LSO. So after silencing the neuronal activity in these 4 classes of neurons, we found that these enhanced responses that you see in otherwise wild-type controls or in parental controls, these were gone down back to normal response. So what this data suggests that perhaps the intact and active LSO neurons are the ones where the modulation is taking place, and they are the ones that regulate high salt intake as seen in case of GR5A and wild-type flies or parental controls where the LSO was intact, the neuronal activity was intact. And as soon as you knock down the activity in these LSO neurons, it leads to reduction in these enhanced responses back to normal in high salt fed flies. Since we are using 24-hour starved flies in our assay, silencing neuronal activity of the LSO by tetanus toxin could not inhibit further feeding on salt. And also remember that there are multiple receptors which are present in LSO. So maybe, we cannot really see the responses lower than this. So to test if it is true also in case of bitter neurons as well, we look different categories of bitter neurons. So first we tested GR33A, something very similar like GR5A, which expresses in the labellum but not in LSO. And in this case also we found that we see an enhanced responses for NaCl fed flies at 200 millimolar. A similar result was observed for 93A. But when we use GR66A, which actually presents in LSO, these responses were gone down back to normal, again suggesting that no matter what receptor these neurons are expressing, the starvation state and the activity both are required in LSO that modulates the salt and salt or taste behavior in flies when they're pre-exposed to high salt conditions. But interestingly, what we found here that in case of GR66A, the aversion to high salt condition, even in normal media conditions, were enhanced in both the conditions, suggesting that perhaps GR66A in general as a receptor and neurons are required to sense high level of salt in the media. So next we wanted to make sure that the sufficiency is coming through the LSO neurons. So for this we tested GR2A and POX neuron GAL4, which specifically express only in the LSO and nowhere else. And as expected, what we found that in both the cases, when the activity was shut down, we did not see any increase at low or high concentration of salt, as shown by feeding assays or by doing spectrophotometry analysis. So again, our data suggests that detection of high salt and feeding regulations are regulated by the for high salt is regulated by LSO. We have also tested IR76B, which is actually a receptor to identify and detect low in salt conditions for fly. But even this is present in LSO, and knocking down the activity of IR76B in LSO did not show any response, just like to our GR2RA or POX-GAL4. It also suggests that the pathways that detect high and low salt in flies and the pathways that regulate the feeding operates differently, and these are not the similar pathway. So in a summary slides, what our study present is an attractive population of neurons that regulates a key aspect of feeding behavior in decision making when flies are pre-exposed to high salt diet. Neurons like LSO, which are shown in the pharynx of flies, present a regulatory system that has evolved to fine-tune our calorie intake with energy metabolism, and these microcircuits are even conserved in humans to regulate the food intake. Our study also suggests that dietary high salt modulates and reshapes salt and other taste curves to promote overconsumption of food in flies. Multiple taste receptor neurons and pathways are involved, as we have seen in our study, but how high salt activates different gustatory receptor neurons is not entirely clear yet. Genetic suppression of LSO neurons inhibits excessive salt intake, demonstrating that acute regulation of feeding behavior by LSO neuronal population in fly. We still don't know the mechanism. It could be due to neuromodulation, change in sensitivity, or impaired taste function, or it could also be that the energy state is very different in flies, which are feeding on high salt, or it could just be a hedonic regulation. Doing calcium imaging and electrophysiology from these areas is very difficult because there is a constant movement in LSO and we see a contraction in this area. So what our study suggests that this is the first time we are showing that a role of activity together with internal state modulates the high salt behavior in fly by LSO neurons. And now what we are investigating are the neural circuits which are involved in the brain, and these circuits could be the ones which are changing the sensitivity towards the high salt. We still don't know if some salt metabolic sensors are also involved in such a phenomena. So overall, our results suggest that pharyngeal taste neurons like LSO play an important role in sodium homeostasis in animals or flies. The linkage between salty and sweet taste modulation may optimize sodium and calorie intake in animals. Our study also provides a mechanism to explain why high salt diets are associated with obesity and emphasize how non-caloric substances can influence the risk of obesity and diabetes. Therefore, and emphasizing that factors like salt should be considered in promoting weight loss and preventing metabolic consequences. So with this, I would like to stop and thank people who are the real force behind all this data, Shivam Kaushik and Rahul Kumar. Shivam and Rahul, and also my funding and my collaborators, RCP for gender support and instrumentation facility, various people who helped in getting us flies. And last but not the least, people who are motivating us to, take our science from bench to society. And we are grateful, that we are able to do this for them. Thank you so much. And I'll be happy to take further questions.
Okay, thank you, Dr. Sharma, for the fantastic talk. We've got quite a few questions. So the first question is from Krishna Melnatur. They say, lovely talk and work. And the question is about taste coding in SEZ neurons. Do you think the tuning of taste SEZ neurons is similar to or distinct from tuning of peripheral sensory neurons?
So, we have some data by other labs. So far, it looks like that certain factors which are working at the periphery, they're also working in the central neurons. But we still don't know a lot of things. So I really can't say that it is 100% like this. But yes, to the extent that we have seen so far in the field is the similar tuning.
Okay. And Abhilasha wants to know, what is the natural source of salt for wild Drosophila populations?
Yes.
He's curious about the range of concentration used in these experiments and how does that compare?
Yeah. So generally in nature, flies, they move to, fermenting fruits to get sodium in nature. And in the lab, we generally add yeast, which is lower than 10 millimolar. So generally, the concentrations that we have used for our assays are very high. So 10 is the same which we use in yeast, and that has been tested also in the curve that I had shown you. But otherwise 200 millimolar is just for our experimental purpose. It's way too high in general.
Okay. Thank you so much. In the interest of, yeah. So thanks, Dr. Sharma. Okay.
Thank you so much.
Thank you. So it's now time for the our third invited speaker of the day. Please extend a warm welcome to Dr. Sonal Nagarkar Jaiswal. She's a senior scientist at CCMB-CSIR, where she and her team work on identifying the mechanisms and the molecular players underlying neural stem cell self-renewal and differentiation, using fruit flies and human neuronal stem cells as models. She's a Wellcome Trust DBT India Alliance Intermediate Fellow and has received the Ramanujan and Ramalingaswamy Fellowships in 2018. So today she is representing the neuronal development field and will give a talk on using and developing novel in vivo tools and techniques based on Minos transposon strategy and CRISPR-Cas9 to study development and maintenance of neural stem cells in Drosophila. So Dr. Jaiswal, thank you for accepting our invitation to talk in this conference. And yeah, the floor is all yours.
Thank you, Anupama. First of all, I would like to thank the organizers. Can you hear me?
Yes, we can hear you and we can see you. Yeah.
So first of all, I would like to thank the organizers for organizing such a wonderful conference and also giving me an opportunity to be a part of it. And today I will mostly talk about my scientific journey from my postdoctoral lab to my current lab that I established in June 2018 at CCMB. So what I'm going to do is I'll tell you about a couple of techniques that I helped establish during my postdoctoral work. And then I'll show you how I use these to basically establish my own research plan. So we all know that innovations in genome editing techniques are much desirable because of, for basically understanding the molecular mechanisms underlying any biological process. And as these tools got sharpened, they accelerated the discovery rate. And indeed, success of a model organism is directly proportional to ease of genetic manipulation. And one such model organism which I work on is Drosophila melanogaster, which has been at the front of discovery due to its genetic wealth and increasingly growing tools, genetic tools for making the manipulation easily. So it started in 1916 with the discovery of white mutant and chromosome non-disjunction that proved chromosome theory of heredity. And since then, Drosophila has been an excellent model system to study basic mechanisms in development and also in diseases. So the major reason for this is that about 60% of protein coding genes of flies are conserved in human. Moreover, 70% of all human disease causing genes are also conserved in flies. So what I'm going to do is I'll start with a very basic method that people used extensively to study gene disruption in Drosophila, which is the P element. So here is a typical structure of P elements where we have a transposase gene, which is flanked by two inverted repeats. These inverted repeats help this transposon to insert into genome randomly. One can use the already established ones, P elements and piggyback, or you can develop a new transposable element depending on the application. So what you can do is basically, or one can do is simply replace the transposase gene with any DNA that you need to put into genome. And these are basically, this DNA is flanked by two inverted repeats, so which will help it insert into the genome. Now you can insert it into the genome randomly with the help of transposase, or we can do targeted insertion now using CRISPR-Cas9 system. So one such artificially engineered transposable element was developed in my postdoc lab, in Hugo's lab. That was before CRISPR. So what do we have here is the system is called MIMIC, which stands for Minos Mediated Integration Cassette. It depends on Minos transposon, which is basically a naturally occurring transposable element in Drosophila, Hydi, and other species. So what we have here is the DNA which codes for transposon is replaced by a series of elements. First is splice acceptor for splicing, followed by 3 stop codons for 3 different phases of DNA. Then we have a poly A tail SP protein and a GFP coding sequence and a yellow body color cassette, which will help us screen flies that have already these insertions. And these are all flanked by 2 HP sites and the MINOS inverted repeats. So why do we have to choose MINOS transposon? Why not P element or piggyback? So the P element and piggyback, they have insertional bias, mostly P element that tend to insert at the 5' region of a gene, while piggyback, we have seen that piggyback inserts within the gene also, but compared to MINOS, it is less. So MINOS transposable element actually can insert at different location within the gene throughout the genome, including the coding intron. So the coding intron we call an intron that is flanked by 2 coding exons. So what we did, we took this artificial P element, transposon, and then inserted in Drosophila in 1 chromosome and then hopped it to other chromosomes. And this way we generated about 17,000 mimic lines, and we got them whole genome sequenced to find out the location of insertion. And about 7,415 we are publicly available now. So as you can see, what we expected, the number of, like a large number of insertion also there is also in the coding region, in coding exon and introns. So now, depending on these, insertion position, we can use these lines for many different applications. Like if there is an insertion which is at upstream of a gene, we can use it to create ectopic enhancer or insulator. If it is in 5' UTR region, then we can use it to create binary factor like GAL4 system. I'll explain it in a few minutes. So GAL4 system or QF system or Lexase system. Then we have insertions that are in intergenic region. You can use that for insertion of transgene or FRT or Cre sites. And then we also have the coding intronic region insertions that can be used for various applications. So most of the methods that I have worked on are based on these coding and intronic insertions. I have listed them here and I'll focus on them today. So I'll focus on first 3 of them today. So how do we use this coding intronic insertion? So first thing is they actually work as a gene trap, wonderful gene traps. How it happens? So this is a normal scenario where you have gene, you have UTRs and poly A. So when the transcription is happening, you have full transcription, and upon splicing and translation, you have the protein product. Now, what happens when mimic is inserted in a coding intron, which we see here? So we call this orientation as a gene trap orientation because the mimic can insert in either orientation. So this is the gene trap orientation where we have splice acceptor followed by stop codons. So now while transcription, the transcription machinery will encounter this poly A tail and it will prematurely terminate the transcription. As a result, we have a terminated or a short transcript. And upon translation, because of this splice acceptor, these stops are introduced. So basically, we truncated this gene depending on where that mimic is inserted. So we created, as you saw in my list, that we created about 3,000 coding intronic insertion that cover 1,800 genes. So we have almost created mutants for 1,800 genes. So do they work? What I did basically for that, I selected 200 random insertions, coding intronic insertions, and looked at them and picked 50 genes that are essential for survival, already known. And then I did complementation test with known mutants, and I found these gene traps are actually strong loss of function alleles. So now we can go and take any of these genes and look at the phenotypes. So this is about the gene trap. We can also use these coding intronic insertion for tagging protein endogenously. How do we do that? Now we have these set of mimic lines where the mimic is inserted in coding intron in either direction, a gene trap or a non-gene trap insertion. We can use both of them. Basically what we do, we exchange the DNA which is present between these 2 HB sites with any DNA which is present between 2 HB sites with the help of 5C31 integrase. Here what I have is a protein cassette, a tagging cassette, where we have a couple of tags flanked by 2 linkers. these are quadruple of GGS. So it gives a flexibility, like some flexibility to the structure. Now what happens once it is inserted? You got a splice acceptor and a splice donor flanking tag. So upon splicing, the tag sequence will be inserted into the mature transcript as an artificial exon. And now upon folding, since the loops are flexible, they, in majority of cases, form a loop and the tag is looping out, which does not interfere with protein folding. So it's an internal tag, not a C-terminal or N-terminal tag, but it also helps in keeping the biological functionality of this protein. Okay, so this thing can be done in two ways. First way, you expand these flies in thousands of number, then you collect eggs, and then you inject this plasmid along with a source of 5C31 integrase. And then you have to screen a lot of flies, hundreds of flies to come to this point basically. Or the second thing is I generated a toolkit where I created fly stocks to do this thing in vivo. So where you have a source of this mimic line and then two other fly lines that will have these tag and the 5C31 integrase. And in 3 generation, you can actually tag any gene endogenously using these 3 flies with the mimic. Okay, so using this method, we actually tag a wide range of protein like membrane protein or transcription factor, ubiquitin ligase, and many more. So what can we do with these tag lines? Everybody can, say we can look at protein expression and dynamics and subcellular localization. We can find interacting partners, or we can do immuno-EM and stuff. But there is one more thing that we can do, which is tag-mediated conditional knockdown. And here I'll talk mostly about protein knockdown. So for that, we need 2 components. One is the US GALFO system, and another one is a degrade FP system. And I'll explain you both. So what is US GALFO system? As we heard yesterday, GALFO is a transcription activator. It'll go and bind to the US sequence, where it will drive the expression of whatever is there downstream to code. Now we can control the expression of this thing which is present here in 3 ways. First, we'll do temporal and spatial control of expression, and that we can do using a tissue-specific promoter or gene-specific promoter. So that will give us spatial and temporal expression, but we can also control the expression level and how that happens. So when these lines were being generated, the people who did that, they actually did one very nice thing. They put HSP70 basal promoter just before the GALFO coding sequence. And this HSP70 basal promoter responds to temperature. So if you keep flies, normally we grow flies at 25, but if you keep them at 18 degrees, the expression of the downstream gene will be low. Or if you shift them to 28 or 29, the expression of this downstream, excuse me, will be high. So now we have spatial, temporal, as well as level control. So I use this along with the degraded FP system. So what is degraded FP system? Is actually modified ubiquitination system, where E3 ligase, which is slim in Drosophila, where you have slim E3 ligase, what it does is it recognizes and recruits the substrate and the other machinery, which then ubiquitinates the substrate and target it for degradation. So in degrade FP, now this slim protein is fused with the single chain nanobody at N-terminal. And this we can express using US GALFO system. And we'll express this in the background of mimic tag line, where every protein is tagged with GFP. So whenever we express this, it'll go and bind to the GFP tag protein and target it for degradation. And as I told you, we can control the spatial temporal in a spatial and temporal way, and also the expression level. So this system, does this system work in Drosophila? So we did many experiments. I'll just show you 2 examples. The first one is, here we tagged DLG, disc large with GFP. And this is the control. This is a Drosophila developing brain, which is still attached to the body wall. You see the expression. So GFP is the DLG GFP expression. You see the brain here. When we knock down DLG GFP using degraded FP and tubulin GALFO driver, so ubiquitous knockdown, what we see, the protein level is down. And we also see there is a phenotype. The brain lobes are very big, which actually is shown for this mutant of these genes, the strong loss of function of alleles of DLG also show the same phenotype. So we say that our system also phenocopies the loss of function of DLG. So this was during development. Can we look at developed animal? So for that, we chose one gene, which is called DUNS. DUNS was already known to be involved in learning and memory. So we selected one mimic line, which covers almost all the isoform DUNS, and we tagged it with GFP. And we show for the first time the protein expression, proper protein expression pattern of DUNS here, which is expressed in mushroom body, which is the learning center of Drosophila. And what we did, we created flies where we brought DUNS GFP, degrade FP, and a mushroom body galpo together. Now we let these animals develop at 18 degrees until the adulthood. So the degrade FP is very low. And then we shifted these flies to 28 degrees to increase degrade FP amount. And you can see by day 4, there is hardly any protein left. And then when we shift these flies back to 18 degrees for 2 days, and the protein is restored. Okay. So how does it affect the learning index? Here we see the first 3 are controls. And then the 4th one is basically when we knock down DUNS GFP with degrade FP at day 4. And you see the learning index has gone down very much. Now, when the same flies, I'm saying the same flies, when we shifted the same flies back to 18 degrees for 2 days, and then we perform learning assay, and we see the same flies now became smart. So they started learning. So you see how good the system is here, that you can use, this mimic system not only for creating mutants, but you can tag the genes and you can do several different applications. So why do we have to think about creating more lines? Because it works beautifully for many different things. So while we were doing this, we realized while we were generating these fly lines, mimic lines, we realized that we were reaching saturation. Means we were getting insertion in same genes again and again. So we stopped at the 17,000 lines, and then we decided to go ahead and use the CRISPR system because it was out there. So what we did basically created, so what I did basically, I designed a construct which we can put with CRISPR system. So we just clone homology arms left and right and inject this construct, which is same except for the marker here, which is an eye marker here, along with Cas9 and the gene specific guide RNA. And you can just insert it in any gene, get it sequenced, get it checked. And then once your line is ready, you can again flip it with GFP tag cassette or any other different cassette that I have generated. So this way you can target each and every gene or most of the genes in Drosophila. So now I have these methods available. Now I have the whole resource available. What can I do with this? So I started when I was towards the end of my postdoc work, I started looking into my interest, and that is to understand basically neuronal stem cell maintenance in Drosophila. So what I did is, so I wanted to use all my resources to find out new genes. So we all know that neuronal stem cell basically asymmetrically divide, and they give rise to an intermediate progenitor cell, which then divides symmetrically and gives 2 neurons or glia. So this happens until the development continues. And once the development is complete, then these some of these stem cells, they undergo quiescence, where they hardly divide or very slowly. But if there is an injury, these stem cells and quiescent stem cells, they undergo, they get, they exit from quiescence and they divide rapidly to replenish the system. So we see that these processes like, self-renewal, differentiation, and quiescence, they're highly regulated processes. And if there is any defect in these, that could lead to great problems. Like if, for example, neuronal stem cell, instead of dividing asymmetrically, it divides symmetrically, or the quiescent neuronal stem cell, abruptly starts dividing, or it just exists before the time is there, or these differentiated cells, they decide to differentiate back to stem cells. This all could lead to brain tumor. On the other hand, now if the stem cell decides to differentiate prematurely, not undergo several rounds of division, this problem could lead to developmental defect, neurodevelopmental defect. So to understand these processes and how they are regulated, the first thing is to find out which are the molecular pairs that are involved in these processes. So to do that, I again used Drosophila as a model system, and I performed two screens. So I utilized the resources that I generated. The first one, at that time I had 1200 independent fly lines, like 1200 genes stacked, and I had generated another resource where I created endogenous GAL4 drivers. So what I did, basically, I took the gene sequence out and inserted GAL4 sequence. So we have GAL4 expressed under the so the regulation is through the gene's native regulatory element. So this way I use these two resources, and first screen I did is I just look for protein or gene expression. So in case of EGFP tag lines, I did immunostaining with anti-EGFP and anti-Miranda, which is a neuronal marker. And in case of these GAL4 lines, I just use USGFP as a reporter. And I found from all of these, I selected 30 genes, new genes that are expressed in neuroblast. So the reason it is just 30 is that I wanted to select the ones that are evolutionarily conserved. So this was my first screen. Then I did another screen, like secondary screen, where I knocked down these genes, either using RNAi for the GAL4 lines or protein degradation using degrade-FP method. So after this, I picked the ones that are that show abnormal brain development. So I identified 13 potential genes whose loss lead to basically abnormal brain development, means either the brains were small in size or they were large in size. So what happened to the others? Like the rest of the 17, they are expressed, what happened to them. So what I know is, while I was doing this screen, I had this data first round. I found out 2 of the genes from my list actually were shown to be involved in differentiation into specific neurons. So the other 17 could also be involved in some kind of differentiation pathway. So they do not affect the brain size, but they might affect the generation of specific neurons. So currently we are, so in my lab, I'm working on 3 of these candidates from here. And today I will just talk about one candidate. This candidate has been linked to microcephaly in humans, and it is a totally uncharacterized gene. So I got it from the endogenous GAL4 screen. And this is a typical pattern that we see when a gene is expressed in neuroblasts. So this is the candidate that I had. It showed the similar pattern. When I knock down this gene using RNAi, I see that the brain was small. So this is the wild type, like a control brain size, and this is almost 1/3 or half in case of lockdown of this gene. So what is this gene? Briefly, it's uncharacterized in all these model system. Any of the model system, it is not known what it does. So cellular process and molecular function is not known. What we know is it's a very small gene. It has two potential transmembrane domains. One is at N and one is at C. So we're looking at it, how it is involved in maintenance of these neuronals, neuroblast maintenance. So what we did, we created mutant using the same CRISPR and mimic strategy, mutant for the gene. We also obtained U6 guide RNA for gene knockouts, and then we obtained an RNAi line for gene knockdown. So in all these 3 conditions, we find that it leads to loss of this gene, leads to larval lethality. So basically, this gene is essential for survival. We looked at brain. So overall, the animal size is also small. Since it is linked to microcephaly, we are more interested in looking at what it does in brain. So we see that the same age animals, you see the brain size is very small than control. And then we looked at carefully what is happening. So first thing we did is we looked at the neuronal stem cell, which are called neuroblasts, so neuroblast markers. Here, the top panel you see in control, green is Miranda, which is a basal cortex protein. And it actually anchors another protein called prospero, which is a sulfate determinant. So you see nicely Miranda is anchoring or localizing the prospero to basal cortex. And we have APKC, which is an apical polarity marker. So in control, we have nicely established apical and basal polarity. While when we looked at mutants, we hardly see 1 or 2 cells where we see, somehow there is a kind of polarity establishment. The majority of cells, they do not have. Moreover, we see the cells that have lost polarity. In those cells, while the Prospero in control is the basal, located at basal cortex. Here it reached to nucleus, which then will drive these neuronal stem cell for differentiation into neurons. So while we were looking at this, we also realized one thing, which is these neuroblasts are binucleated or multinucleated. So you see in control, every neuroblast has a single nucleus, while here you see either 2 or sometimes 4 or sometimes 6 neuronal stem cells. So clearly it shows that loss of this gene function leads to cytokinesis defect. Now, the cytokinesis defect can be majorly because of 2 reasons. In our case, I'll explain you. So this is normal cytokinesis. What happens during cytokinesis? First, ectomyosin ring is assembled. So in a polar cell, ectomyosin contractile ring assembles on both the sides, and then it starts to contract basically. While this is happening, the furrow is increasing. There's another process happening, which is small vesicle, they bring membrane components and they come to this leading edges basically, and they fuse with this increasing membrane. And towards the end, then there is an abscission, and then these 2 cells separate. So what I said, that basically first thing is happening is assembly of ectomyosin contractile ring. So it is possible that in our case, since we don't see polarity, maybe this ectomyosin contractile ring is not assembled, because polarity is important feature for this assembly. We can check that using myosin 2, which is spaghetti squash in Drosophila, or septin proteins. They also assemble here, which is peanut. The second thing which could happen is perhaps these small vesicles, which are supposed to bring the membrane components, maybe they are not reaching this side, or they are not fusing to the membrane. So the reason I'm saying that perhaps the vesicle transport is involved, because this gene has 2 transmembrane domains, and it has been predicted to be involved in first to localize to ER and to involve in ER to Golgi transport. But we don't have any experimental evidence right now that what is happening. So this also we can see by using some vesicle markers like RAB11, syntaxin, COP1 and COP2. So what we did, we had antibody for RAB11 in lab, and we did a quick experiment, which we now confirm. So here in left side, you see green is phalloidin. So it's marking the wall, like basically cell membrane of neuroblast. Then we have RAB11 in red. So you see there are some puncta of RAB11, but when you look at mutants, we see the RAB11 is like at least 20 times higher than the control. And what majorly we see is, so this is one section, but what we see on both the side, there are these 2 nuclei, you see 2 nuclei here. On either side, we see a big blob of RAB11. And this is there in almost 90% of neuroblasts. So we do see there is an accumulation of RAB11. So we think that it could be that the vesicles are coming and they're getting accumulated. Either they are not getting fused because there is no leading furrow, or there is a leading furrow, but they have problems, excuse me.
Dr.
Infusion.
It would be, we are kind of a little bit over time, so it'd be great if you could.
I'm done. I'm at the last slide.
Thank you so much. Yeah.
So this is what, so just to summarize, what we think right now could be happening is that loss of this gene leads to vesicle transport defect. And that because of that, we have defect in polarity, and both of these could lead to cytokinesis failure. At the same time, so we see multinucleated cells and these cells that have lost polarity, and which totally leads to loss of neural cell population and leads to small brain phenotype. So currently we are working on all these, both of these directions, and we'll see what happens. So just to quickly summarize, that loss of this gene leads to developmental delay, larval lethality in small brain due to defects in cytokinesis and loss of polarity. And we see that there is defect in endocycle vesicle recycling. And with this, I would finish. And it's Aishwarya who is doing the main work. And these are the other members of my lab, including technicians and dissertation student. And yes, and I would actually thank you guys for attending the talk. Thank you.
Thank you, Dr. Jaiswal. It was very nice, elegant genetics and interesting results. Yeah, so we don't have time to take questions now, but we have a few questions. Could you, it'd be great if you could answer them in the chat, in the question and answer box.
Okay.
Thank you. Yeah, and thank you so much for
Thank you.
accepting our invitation today. Thank you. Okay, so
Unshare it. Thank you.
Yeah.
So I'll just write the answer here. Type the answer here.
in the question, yes, in the question and answer box.
Thank you.
Thank you. Okay, let's just take a break for a couple of minutes while we help our next speaker set up.
Okay. Anupama?
Yeah.
Okay. I can take it on from here.
Okay.
Okay. Thank
Yeah,
you.
thanks a lot. Yeah.
Okay. Hello, everyone. I'm Sulagna Das. I'm a research assistant professor at Albert Einstein College of Medicine in New York. So I'll be taking over from Anupama here and moderating the rest of this session. It's my great pleasure to introduce Dr. Poonam Thakur. She's an assistant professor at IISER TVM, where she and her team works on developing better mouse models of Parkinson's disease in order to decipher disease mechanisms for developing biomarkers as well as therapeutics. She's a recipient of 2019 Wellcome Trust DBT India Alliance Early Career Fellowship and is a member of various societies like the International Parkinson's and Movement Disorder Society, the Indian Biophysical Society, and the Indian Academy of Neuroscience. Today, she is representing the neuronal disease field and will give a talk on understanding the mechanisms underlying the differential vulnerability of neurons to alpha-synuclein aggregation, which is one of the main culprits in Parkinson's disease. Dr. Poonam, thank you for accepting our invitation to give a talk in this conference, and the floor is all yours.
Thank you so much, Dr. Sulagna, for the kind introduction. Are my slides up and visible?
Yes, we can see them and we can hear you perfect.
All right, thank you. So today I'm going to talk about how alpha-synuclein fibrils disrupt the firing activity of dopaminergic neurons. So this whole work is done in context of Parkinson's disease. And Parkinson's disease is what many of you would already know. It is one of the most common age-associated neurodegenerative disorders and affect millions of people worldwide. Unfortunately, to date, we do not have any cure for this disease. Patients who have PD suffer from a variety of non-motor symptoms, such as problems with sleeping or their bowel movements or sense of taste and smell, smell. But this disease is majorly associated with the motor symptoms. And this encompasses a large range. For example, these patients often have these masked appearance. They suffer from tremor, rigidity, and problems with walking, and posture cannot be maintained, strict, rigid. So all of these problems are what patients faced. But here, I would like all of you to take a closer look at this picture and see that here we are seeing a man in this cartoon. And I have been wanting to put a female who was also representative of these motor disorders that are happening during Parkinson's disease. And unfortunately, despite my extensive search, I could not find a single picture to put in a conference that is actually celebrating female neuroscientists. So this just tells you how deeply this, inherent bias runs in the field of sciences, where not just representation of scientists is cut off, but even the representation of the societies that we serve is not enough. And in this scenario, what Vaishnavi and Shruti have started, I deeply appreciate this initiative. And I really would like to congratulate them to bring this conversation and bring this important issue into highlight. And I'm very, very immensely happy to be associated with them to organize this conference as well as showcase my work here. All right, so returning to the Parkinson's disease now. So all of these motor symptoms that show up here are majorly caused by the loss of these dopamine producing neurons in brain region called as substantia nigra. Here is a human brain section which is showing these dark colored neurons, which are dark due to presence of neuromelanin pigment in a normal person. And in a person affected with Parkinson's disease, you can see a severe loss of these neurons in comparison to a control subject. And this loss of dopamine results in all these motor symptoms because this leads to lack of dopamine release in the basal ganglia circuitry, which controls the motor movements. And this loss of dopaminergic neurons is actually believed to be caused by the appearance of these aggregates which are rich in protein alpha-synuclein. Alpha-synuclein usually is a small cytosolic protein. It is natively unfolded, but during certain conditions, it can aggregate and form these beta sheet-rich aggregates, which can also then accumulate many other proteins, leading to these complexes of multiple proteins and dead organelles, damaged organelles called as Lewy bodies. And the Lewy bodies, when they show up in the neurons, can lead to their death. However, the case is not as simple as that. In this picture, you can see the Lewy pathology severity during Parkinson's disease. And you can see, other than substantia nigra, there's a large number of regions which get affected with the disease, including olfactory bulb, hippocampus, and even areas of cortex during the later stages of the diseases. However, the amount of Lewy pathology is not directly proportional to the amount of cell death you see in these regions. So here you can see that substantia nigra region is disproportionately affected by this Lewy pathology in comparison to any other brain areas. And this is what scientists have been trying to find out for years, that why is that dopaminergic neurons are so vulnerable to Lewy pathology. Now, several hypotheses have been put forward to explain this difference, and a major hypothesis that has been well accepted in the field for several years is that dopaminergic neurons are very different from others in some of their properties. First, they have this giant axonal arbor in comparison to most of the typical neurons that are present in the brain. In fact, the axonal arbor of dopaminergic neurons is one of the largest in the central nervous system. On top of this, these axons are also non-myelinated, and the number of synaptic sites these axons form is immensely high compared to any other neurons. So this one cell body has to go and feed all of these axonal and synaptic sites. So imagine the metabolic burden that comes on these neurons. And this is not all. Okay. We know all the neurons fire when they get some input from the synapses, and then they can fire some spikes. However, these neurons also have another feature, that they display this what is called as intrinsically active firing. Okay, so this happens without any synaptic input. This is completely generated independently within the cell, and this is called as pacemaker firing because it is slow and rhythmic in nature. So this is a term that is borrowed from cardiac physiology. And this means that with each action potential that is firing in these neurons all the time, a lot of calcium is coming in. And then cells have to deal with this constant influx of calcium. So again, all of these factors add to put a very high metabolic stress on these cells. And this is what we call as a stressful pacemaker theory. So while this can explain to a good extent why these particular neurons are more vulnerable compared to other neuronal subpopulations, it does not explain the differences that we observe within the dopaminergic neurons. So there are dopaminergic neurons in several nuclei within the brain. So there is a region called as ventral tegmental area, Just for ease, I'm showing this in brain atlas, so it is more clear. So this is the ventral tegmental area, and this is medial substantia nigra region, and this is the lateral substantia nigra region. And it is a well-known fact that has been observed in all preclinical models as well as in humans that substantia nigra neurons are far more vulnerable to cell death in comparison to VTA neurons, which are known to be much more resistant. Even though they are both dopaminergic, they all have the giant exons, and they all have pacemaker fibrils. In fact, even within substantia nigra region, it is the lateral substantia nigra regions that are known to be more vulnerable in comparison to the medial substantia nigra neurons. So it is clear that all dopaminergic neurons are not same in terms of their vulnerability. So then there must be some additional factors that must be describing these differences in vulnerability that are beyond the stressful pacemaker theory. And this is where my work was focused on to, because I wanted to understand what is the reason. And for this, we decided to take an electrophysiology approach. But the first hurdle on our way was how to identify these different cell populations within the dopaminergic neurons. If we are just studying all of them as a whole, it's easy to just have a transgenic line in which these cells are labeled with the fluorophore, and we can easily identify. But as of now, we do not have any molecular markers that can help us to distinguish between various subpopulations of dopaminergic neurons. So in that case, what we did was we relied on using their axonal projections. Okay, so these neurons here in VTA and substantia nigra region send their axons into the striatum region in a very topologically organized manner. So relying on these projections, what we did was we did retrograde labeling. So we injected these fluorescent retrobeads into particular areas of striatum. One is nucleus accumbens course, which is sending its projection back into the VTA region. One was dorsomedial striatum, which you can see here is sending its projections back into the medial substantia nigra, and other was dorsolateral striatum, which is sending its projections back into the lateral substantia nigra region. So by this selective labeling of neurons, we were then able to identify these various subpopulations. 2 to 3 days after this fluorescent retrobead injection, we sacrifice a mouse, we obtain brain slices, and then we perform patch clamp recordings. To see how the activity of these neurons is changing in response to alpha-synuclein, we decided to apply alpha-synuclein individually on a cell rather than on a whole, as a whole on the slice, because in that case, we cannot be sure which neurons are taking up these fibrils and which are not. And a lot of separate dynamics will kick into the picture. So to cut down on that complexity, what we did was we put in these fibrils within this recording pipette solution, and we approached the cell, we opened the cell, and then alpha-synuclein could slowly dialyze within these cells. And while recording, we also put this neurobiotin dye within this solution. So all the neurons that we record are post hoc identified. So each neuron that is recorded is labeled with neurobiotin. We confirm that it is positive for tyrosine hydroxylase, a marker for dopaminergic neurons, and that it also contains these red fluorescent beads. So identity and specificity of all of these neurons that were used in these recordings was well established. So once this protocol is in order, the first neuron population that we looked at was a VTA neurons projecting to nucleus accumbens core. Here is a histological section in a very small magnification. You can see a few green cells, which are the ones we have recorded. And here are the whole cell recordings of these neurons for 300 seconds. And this is recording without any induced current. So this is just the spontaneous recording, which is arising from completely within the neurons. So you can see adding these alpha-synuclein fibrils did not cause any change in this firing pattern in comparison to the control group. In addition, we also use monomeric alpha-synuclein, which is the physiological form of alpha-synuclein, as a control to see that it is not just the presence of protein amount in the recording pipette that is causing any alterations, if at all. For those who are not very used to seeing electrophysiology data, you might be wondering, like, I'm not seeing spikes. So just this is a stretched out picture of first 10 seconds of this recording. So you can see all the individual action potentials here. Because this is a bit compressed, so it's a bit hard to see the individual spikes in this case. All right. So this is the quantification of this data. We have plotted the firing frequency here. You can see there are no differences in various groups over time. We also record average membrane potential. We cannot have resting membrane potential because these neurons, as you can see, are not resting at all. And we are also plotting the regularity of firing or coefficient of variation of interspike interval. And so smaller the number, more regular the firing is, and the bigger it is, more irregular the firing is. And you can see the regularity of this pacemaker firing, which is the hallmark of this type of firing, is also not changed. Next subpopulation we looked at was the medial substantia nigra neurons projecting to dorsomedial striatum. Again, a picture showing some recorded neurons here, and you can see monomers do not cause any change in firing activity. But in case of fibrils, you can see there are some mild irregularities in firing in some places. However, upon quantification, we saw that this firing variability did not appear in all the neurons that we recorded, and because it showed up in only few neurons, it did not cause any statistically significant change in comparison to the control population. Also, overall, we did not see any change in firing frequency or the membrane potential here. But when we recorded these lateral substantia nigra neurons, you can see that fibrils cause a very dramatic change in the firing activity. You can see very, very quickly after this fibril application, firing starts to become irregular, and membrane potential is also becoming more negative, that is hyperpolarizing. And after a while, firing activity completely ceases. And upon quantification, you can see the very dramatic loss of firing activity, loss of membrane potential, as well as a dramatic increase in the regularity of firing. So once we saw this effect, and we could see that lateral substantia nigra neurons were most susceptible in response to alpha-synuclein fibrils, we wanted to ensure that the effects that we are seeing are not just an artifact of, shoving it down their throat in this case, actually. So just to confirm that these effects are actually, physiologically possible, even when alpha-synuclein fibrils are applied extracellularly, we did another experiment. And in this case, what we did was, we are recording these neurons with help of one pipette, but here we do not open the membrane, so that all the fibril intake is happening through an intact membrane and not a membrane that we have opened up. But we are applying these fibrils via a second patch pipette that is held in a very, very close distance to the neuron that is being recorded. We also had a control solution. And here in this on-cell recording mode, you can see that adding control solution does not cause any change in the firing activity. But when fibrils are applied through without opening the cells, even then loss of this firing activity can be clearly observed. So we can also see that on quantification, there's a clear loss of firing frequency as well as increase in firing variability here. So overall, we could see that the lateral substantia nigra neurons were observed to be highly vulnerable to alpha-synuclein fibrils. Medial substantia nigra neurons show mild irregularity in some cells, but not in most of the cells. And VTA neurons did not show any change in response to alpha-synuclein fibrils. So the pattern of selective vulnerability that we observed in the preclinical models or in the humans actually was also replicated in this experiment, which lasted 5 minutes. And this could replicate the pattern that we are observing in humans in a process that lasts decades. So this was, a very good sign for us that, and also indicated to us that this selective vulnerability of these neurons is something probably which is very intrinsic property of these neurons. It is just, encoded within them somehow. And how is then, next question is obviously, then what is the mechanism? Why are lateral substantia nigra neurons so vulnerable to alpha-synuclein? What is happening there? How are they causing all these changes? So this firing activity that is taking place in the neurons is coming with help of number of ion channels that exist on membranes of these cells. And to find out the potential culprit, our eyes went on to KATP channels. Why? So these KATP channels are, they are redox sensors of the neurons. Under normal conditions, they are always closed. So they are composed of these two types of subunits. One is this pore-forming subunit called as KIR6 here, and other is the sulfonylurea receptor subunits, which are regulatory subunits here. And under normal conditions, they are bound to ATP here and remain closed. But in conditions of oxidative stress, or when cells have some energy crisis, the amount of ATP in the cells go down. And as ADP amount rises in the cells in comparison to ATP, this ADP will come and bind here, leading to opening of this pore, leading to extrusion of potassium from the cells and causing hyperpolarization of these cells. So what this acts as is actually a molecular brake on the cells. So in case there is a lack of ATP or there is a very stressful condition and cells need to route their ATP production to more important survival processes, so the firing activity can be temporarily halted and neurotransmitter release be stopped in these conditions. So if you remember, with in the lateral substantia nigra neurons, we saw a very clear hyperpolarization of the membranes. So we suspected that maybe KATP channels are opening up in response to the alpha-synuclein fibrils. So to confirm this hypothesis, what we did was we performed another set of recordings with presence of glibenclamide, which is a selective inhibitor of KATP channels. And we could see that glibenclamide application prevented the loss of firing activity that we observed in presence of fibrils. And this was also quantifiable. We could see the clear differences. Next, to further confirm the involvement of KATP channels, we took some transgenic mouse in which KIR6.2 subunit was selectively fluxed out in the dopaminergic neurons of these mouse. We also had corresponding wild-type littermates as controls. And here we could see that in controls, we could again see the loss of firing activity with fibrils. But when we have the mouse with KIR6.2 flux out, fibrils are no longer able to cause any changes in the firing activity, further confirming the role of KATP channels. Yeah, this is the quantification of the same results here. Okay, so now we know that KATP channels are involved in this opening, but KATP channels open under stress conditions, right? So then what is the stress in this condition, in this case, that has caused the opening of KATP channels here? So from literature, we found out that alpha-synuclein can cause dysregulation of calcium levels in the cells. And when calcium amounts, excess amounts of calcium are inside the cells, then what they try to do is put them into the intracellular calcium stores to try to get rid of them from the cytosol, right? So ER and mitochondria are one of the common intracellular stores. But when a lot of calcium enters the mitochondria, it can lead to ROS production from them, which we thought could lead to opening of KATP channels. So to test this hypothesis, what we decided to do was block this calcium entry into mitochondria. Most of the calcium that enters the mitochondria enters through this channel called as mitochondrial calcium unipotent. So what we did was we just blocked this channel with the help of Ru360, a selective inhibitor of this channel. Again, fibrils are causing loss of firing activity, but in presence of Ru360, we could see absolutely no change in firing anymore. And this is the quantification, so a very good rescue experiment. So just to summarize this data, we are hypothesizing that alpha-synuclein is causing calcium dysregulation, which enters the mitochondria, causes production of ROS, which goes and open KATP channels. And if we stop this process either by blocking entry of calcium into mitochondria or opening of KTP channels, we can help mitigate the alpha-synuclein caused changes in the loss of firing activity. However, we are yet to do many more studies to directly look at the calcium changes in presence of alpha-synuclein, and that is what we hope to do in near future. With this, I would end my talk, and I would like to acknowledge Jochen Roper. He was my, sorry about that, he was my postdoc PI, in whose lab this work has been performed at Goethe University, Frankfurt. My lab mates there and lab technicians who have helped me a lot in this work. Kenvin Luke at Pennsylvania, who provided us with fibrils, and IISER TVM for helping me set up my lab currently, and welcome Trust-DPT India Alliance for funding. And last but not the least, my lab members and my team here. And I'm happy to take questions. Thank you.
Thank you, Dr. Poonam. That was an excellent talk, very well explained into the insights, the molecular mechanisms of the susceptibility of neurons. So there are a couple of questions. The first question is from Abhilasha. Do the DLS ISN neurons survive after the fibrils treatment? And can this effect be washed out, like the effect of the fibrils on these neurons?
Yeah, so Abhilasha, I just want to clarify one thing, that when we are doing these slices, so I mean, once we sacrifice the mouse, the neurons are in that way, kind of dead in a way. We are keeping them artificially alive for the purpose of recording. So only thing is that if we keep perfusing alpha-synuclein fibrils for a very, very long time, at one point cells will absolutely stop firing, swell up a lot, and then burst out. So I tried recording for like 30 minutes or so, but at one point after so much dialysis into the cells, they are not going to, be viable anymore for to give active recording, if that's what you meant by live here. And the second part of question is that washout effect. So that is not possible to do in the cell attached mode, because once seal is formed, we cannot leave the cell and reform the seal on that one. But in the on-cell mode, we did try that, and actually we saw that it was washed out. For the duration that we did, it was small duration, not a long time, but if like up to 2 minutes of blowing fibrils on the cells, and then after that, if I put normal solution, effect could be washed out.
Thank you. There's another question from Shruti asking, were you perfusing the fibrils and other proteins in perfusion over the whole slice? And if yes, did you happen to check what it does to neurons in other places like the cortex or the hippocampus?
No. So actually, I mentioned that in the beginning of my talk, because if we just put them on the entire slice, it is going to be very, very difficult to then know which cells have actually taken it up and which cells have not taken it up. And that would lead to, a lot of complications in our analysis. So what we did was we were applying these fibrils through the patch pipette. So each neuron that we are recording is getting a very, fixed concentration and amount of alpha-synuclein. So no general throwing all the slices.
Okay, so I think we are a little out of time. So there are a couple of more questions, and if you could answer them in the chat box, in the question and answer, that would be great. We just have to move on to our next speaker. So thank you, Dr. Poonam, and I think if I'll move on to our next speaker, who is Dr. Kavita Vemuri, and we would like to extend a very warm welcome to her. She's an assistant professor at IIT Hyderabad, where she runs the Perception Engineering Group. Sorry. Okay. She and her team has worked extensively on various areas of cognitive systems, including research on the visual system to understand color perception and development of low-cost eye measurement systems to measure various eye-related conditions. Also, her cutting-edge research extends to questions related to neuroeconomics and fluid intelligence, and has been designing tools for visuomotor rehabilitation post-stroke and motor-related degeneration. She wears many professional hats, including being the founder and head of the Center for Innovation and Entrepreneurship at IIT Hyderabad, founder and technology counselor of the Technology Development Center at CII Chennai, as well as an assistant editor for University Press Orient Longban, Hyderabad. So, before we move on to our talk, we would like to let the attendees know that this talk will include discussion around topics such as sexual assault and rape. We acknowledge that this content may be difficult to listen to. We also encourage you to take care for your safety and well-being by disengaging if necessary. Today, she is representing the cognitive neuroscience field and will give a talk on how women's socioeconomic status or profession could change people's assessment of and empathy towards them, including responses to rape narratives. Dr. Kavita, thanks a lot for accepting our invitation to give a talk in this conference, and the floor is all yours.
Thank you. Thanks a lot for the organizers for giving me this opportunity. So the previous talks were all about neural firing and cells, and now we go into almost social neuroscience and social cognition. So yes, there are some topics and there are some findings which might be disturbing. So I would like to have a discussion and not go into, my talk, I'm going to reduce the whole duration of the talk. So I want more discussions on it. So once again, thank you for the organizers. Just before I start, I just want to introduce my team. Mohit Goel worked on rape victim narratives, Nikita Sharma on faces, appearances, and empathy, Shibangi on the default mode network as a marker, Shrija is working on sexual objectification, Ayushi is also working on sexual objectification, but also the role of alcohol on the executive control. Priyanka Srivastava is a faculty in our institute, and she comes from cognitive psychology background and helps in all aspects of design. Radhika Krishnan comes from social sciences. So what am I going to cover? Very quickly, I'm going to cover the empathy research that forms the basis for our work on looking at why empathy as one of the most important things for rape victim, and the sexual objectification studies and possible interventions. We'll be covering 2 studies plus a bit of the preliminary studies we have done on sexual objectifications. And we are using the term victim here rather than survivor. This is nothing to, this is not a political statement, but it is more to do with the existing literature which uses the term victim. Okay, so empathy has been studied in my lab for a while now, almost from 2011-12. And my research was on that. We looked, usually empathy is kind of broken up into 3 modes. There's a cognitive empathy, which involves perspective taking, theory of mind, and mentalizing. That means our ability to actually look into somebody, understand what somebody else is going through, just putting ourselves in somebody's shoes. And then there is the affective empathy or emotional empathy. That means we can actually take over some of their feelings, even though we don't make another kind of a difference. And then there is a pro-social concern and empathic motivation. That means it's altruism in all sense. That means we feel these two and then we actually even act on it. Areas of the brains involved in it have been identified to some extent by various studies, starting off from pain empathy, where usually they show somebody being, somebody going through a small pain. It's like somebody being jabbed with a needle, or an image which shows somebody's hand being jammed in the door. And we look at the areas of the brain that light up. In my own research, I've used movies which are naturalistic, longer, and I at that time made this premise that when you give a longer narrative, it basically sets the perspective. It allows you to take the role of the protagonist. So I won't go into, all of you are here from neuroscience areas, so going into actually the areas of the brain and explaining is actually preaching to the choir here. So I'm just going to go forward. So what is our base research, or what was it that we were interested in, and why empathy? So empathy is it's especially related to rape victim response is important, not just as a response that, say, the police give or the security personnel give, or even what the accused feels, the rapist feels. It's also about what we as society actually do respond to when we hear about a rape victim. Okay, things have changed and we are happy for it, but there's still a lot more to go. So we started looking at the empathy response factors. What all factors are possible? I mean, this is just a small subset of everything that was possible, and this came up with after many conversations with the she teams in Telangana, with the activists, the traffickers, everybody that we could think of course, literature survey. But we started focusing on the fundamentals, going into social stereotypes and biases that exist in our society and as societies across the world in different forms towards the female gender itself, not just to a rape victim, but even to the female gender, which could actually kind of normalize a crime like rape. Okay, and also a response to a victim. And toxic masculinity was something that came up with all discussions with men itself, from our from the student community, and it was a very nice and open discussion. And they said that there is this whole sense of the feeling, or the kind of, we have to stand up for something. I mean, we have to kind of be something that the society wants us to be, and hence they kind of normalize some of their behaviors, small or big. Power is much more complex. It could be power of religion, power of political power, power of money, caste, whatever, or just power of just being a male. Okay, and this adds to the rest of it. Media plays is a much more complex role, the positives and the negatives of it. Media can over objectify women, and that could also lead to a lot of problem. Alcohol, this came up because of the number of accused, always also convicted, that we kind of went and met in the police stations, where they said that they were under the influence of alcohol. They really didn't know what they were doing. Okay, so we've even interviewed a rapist who, was involved in a very nasty crime. I'm using the word nasty because I can't use something stronger than that, on a child. We haven't gone into these, but these are very important. That means the clinical reasons for it, the hormonal imbalances that can be kind of the background for any such kind of deviant behavior, and of course the criminal behavior. So before I actually go into presenting why, what our own data, one of my colleague Priyanka would actually present about why rape matters. I'm hoping that you can hear the audio from her talk.
Hi, I'm Priyanka Srivastav. I did my PhD in cognitive science and currently I'm a faculty at IIIT. My research focuses on basic and applied cognition. I have been working in area of cognitive psychology for over 11 years and recently expanded my research focus to emerging technologies like virtual reality. With Kavita, in this project, we are trying to understand the neurocognitive perspective of sexual victimization and rape mindset. Rape is one of the most serious forms of violence and highlights the gap between stronger and weaker section of society, the gender inequality, and the violence of women's individuals' rights. Rape has traumatizing impact on one's mental health and well-being, and more often than not, it leads to PTSD syndrome. The growing rate of rape cases, or at least growing number of reports on rape cases, indicate a serious concern over social architecture of normalizing rape culture. It's not just concerning to India, where rape is perceived as consensual, at least in some part of the society, and where still it is difficult to define marital rape. It is equally concerning to developed countries like US. Recent report on rape culture of US has highlighted the points that fuels the rape culture, concepts like traditional gender roles, hostility towards women, acceptance of violence, to name few that promote rape culture. Important to note that the cognitive mechanism should be analyzed in relation with social and cultural perspective to have a holistic understanding. It's a global issue, and therefore socio-cognitive approach will help us understand on how we perceive rape as an act and the victim. This approach employs individual ability to perceive, attend, memorize, and make decisions. Measures like response latency, differential focus on perceived risk, differential brain engagement, specifically related to perception, attention, empathy, and emotion, while processing the relevant and irrelevant stimuli, will help us understand the individual as well as the socio-cultural rape mindset and normalization of rape as violence. Thank you.
So we started off with this understanding, and we conducted the very first set of studies that we did was on looking at there's a bottom of the pyramid and the top of the pyramid kind of a thing. Where do we start approaching such a complex problem? So we started off at two different levels. So the very first one was, let's understand what is the society's perception of respectability to careers, professions that women actually are engaged in nowadays. And we took that ratings from the first one, and then we conducted a narrative kind of a study. That means we made narratives of an actual rape incident, and these are basically news reports. These are incidents that happen across the world. Standing at a bus stop and something happens, and it's actually a first person narrative. And then we conducted this on civilian population and the police personnel. And then the third, we then did some filtering of the narratives and did an fMRI study of narratives. And we added female faces. These are actually computer generated, but they were the average faces of a typical Asian woman as generated by University of Florida. And then we did photoshopping to make it change its appearances. And the information that we gave was that these women were rape victims. So in the very first one, which was on respectability, that means all we asked was, if somebody in your family, a female member in your family, was planning to take up a career, what is the respectability you would give? And we actually gave them 21 different options. And we didn't even add options like, say, bar dancer or maid or something, but we just gave them this. And as you can see, the bar manager, a model, a receptionist, a clerk, actress score much lower than, of course, the doctor and the teacher. Okay. Yes, we also asked them whether it was based on education. They said partly so, but mostly it is on what society perceives as something as a respectable career, where they have to interact with people. This was with 149 participants, and there were 34 females and 115 male participants. But the most interesting thing was the data below, where we see that the male and female participants, after averaging out for the differences in the numbers, they're almost the same in the respectability score that we are giving. So women actually also have the same social biases in terms of respect to a certain career as men have, and this data shows it. Then there was no significant differences in any of these ratings. So from here, we took a list of a few professions. That means a teacher, engineer. We took a few, and we also added. We removed bar manager and model because they were way below, and we just wanted to take few which were in the almost middle and the higher. And what did we do? We created narratives like this. I don't think we have the time to read it, but they're pretty graphic. At the same time, they had to pass our ethics, so we actually had to control for a few things. But for most of us who read newspapers about articles about rape, this is all typical what you would read almost every day you would read an article like this. Okay, this is a first-person narrative. Now, what did we do? We took civilian population. There were 206. This was most of them were done in the lab, and the police personnel were all handwritten and done in their office, and translations were done of the narratives in their language, Telugu and in English. Okay, they had a choice to take. So, what was the other questionnaires we took? We took empathic concerns. This is the IRI Davis reactivity index, and empathic concern, perspective taking, and personal distress. As you can see, the civilians were slightly higher than the police, but not so high. Okay, we are almost similar. And the interesting thing was, both when were talking to the police post the experiment, they said that the narrative actually made a huge difference to them. So there, if you actually can see, their empathic scores based on the narrative context, when we set a context rather than just giving a statement, their empathic scores were higher than even the civilians. Of course, we kind of are trying to understand why this is so, but one could be just that there are so many media reports of rape that after some time maybe we've just kind of blocked things out. But that being, I mean, not to be taken lightly, we should actually act on this. If you look at the empathic empathic concern scores, these are not just on the narratives, but these are basically, they were questions designed based on the IRI index, but specific to the narrative. We again see that the police are slightly higher than the civilian populations. Perspective taking, yes, the civilian population is slightly higher in some cases, especially when there is a teacher. The condition of no profile is when we didn't add anything. This is almost a neutral condition. Okay, and the civilian population was slightly higher in perspective taken than the police. Personal distress, as expected, the police were low. That's because they listen to so many cases every day. So this was a eye opener for many of us who would say that, okay, maybe civilian population, we are supposed to be more more empathic to rape victims. Police are supposed to be not so. But I think, like one of the top police officials said, that they all come from the same population. We need to kind of intervention measures should be there for both of them. So the third phase of the study was an fMRI study. So we took narratives, these narratives, we had to change them because every participant, were 20 participants, were actually reading two rape victim narratives, one which was for general empathy and one where there was no profile given, but it was still rape victim empathy. Two were with professions. And then we showed them faces of the purported victims and survivors. Okay, we didn't have funding for this at that time, so we just had 20 university participants, and they were all male, because we just wanted to establish and see if we could actually extract the empathy networks first, and we were able to, if we can see the difference in the profession based narrative that we had generated. So the design was very simple. We had a narrative which was for 80 to 60 second text narrative. All the students were able to read. They were graduate students, and there was a gap, and then the narratives go on. They were all randomized so that you don't have bias. So the professions we had taken was a bar dancer. We wanted the extreme condition, and the teacher, and the no profile. Of course, we didn't mention any profile of it, and the non-general empathy was a narrative of a visually challenged person. So this was our objectives. We also had their behavior data. That means they rated not only the narratives, but also on the rape victim empathy score. That is the rape myth acceptance and the rape victim empathy score. So rape myth acceptance is when these are scores generated, I mean, these are used across the world. They kind of give different perspectives. I'll come to it in a few slides. And we wanted to also conduct a comparative analysis between narratives. So we applied both the methods, the JLM method and the independent component analysis method, because our narratives were almost for 80 seconds. So the very first thing was on the behavior scoring that they gave. We also broke it up into empathy, pity, sympathy, and compassion, just in case there was a confusion between actually what do we feel, what do we express, and we also define them as per dictionary. And you can see that slightly compassion, sympathy is pity was kind of not so. This one, if you, this is the box mixers, the quadrant distributions are not so uniform. And if you actually look at the distribution across the 20 participants, you can see a lot of intersubject differences in all these four, and of course the average. So there is a difference, intersubject differences between in the scores. But the sympathy was the one that actually stood out, more sympathy than actually empathy. And this is from the rape myth acceptance. This is a score. And if you actually look at this, the higher the score is, the more the rape myth is rejected. So this is the Illinois newer version. So higher the score means the rape myth is rejected. Okay, that's a good thing. So you can actually see that there's lower score for she lied, which basically puts a doubt on the victim itself. So these questions are very interesting questions, applicable for any. There's no, the questions are generic. It need not have to be customized for the Indian population. And she lied. The acceptance was more because they attributed some sort of a doubt on the narrative of the victim. And so was he didn't mean to. So this was again actually giving the benefit of doubt to the rapist or the accused. And so when we ran also the correlation analysis just from the behavior data, that is their scores on empathy, sympathy, pity, and compassion combined, and on the rape myth acceptance test, you can actually see that she lied and she asked for it had a positive correlation for the three profiles: bar, teacher, and no profile. Again, kind of reiterating that though there was an empathy, which was good because these were 20 graduate students, and also I think the population per se now understands about the problems in the society, but it also still shows the certain social biases that exist, especially when they put a doubt on the there's a benefit of doubt given to the accused, and they doubt the narrative of the rapist. Then the what does the fMRI data analysis show? What we see is for, especially for the general empathy, this is the group level random effects analysis. General empathy, the both the activations that survived uncorrected only because we could not, none of the activations survived or corrected, family wise error correction. General empathy actually stood out, which was also again supports previous studies which showed that people do have, rapists do have general empathy, but what they lack is rape victim empathy, or actually empathy to the one they have raped, the victim of their own crime. Okay, no profession, when you don't actually have noted a profession, it is very difficult to correlate with them or relate with them. But activations survived, more, areas of activation survived. Whereas for the bar, that is the bar dancer, sorry, this labeling, and for the others didn't survive, but bar dancer greater than teachers, only the inferior frontal gyrus. These are, the inferior frontal gyrus is actually associated or supporting the cognitive empathy area. So what is it that, looking at all these results, there were a lot more of the fMRI analysis. I won't get into the details, but most of them didn't survive. That means the bar dancer or the teacher and general empathy did not survive. The only narrative that actually showed higher significance was general empathy. So general empathy was high, whereas rape-related empathy was kind of low. Okay, that's the final takeaway from what we found. So general empathy was high, but rape victim, somehow the empathy towards a rape victim is generally low. That's our final finding. And the ICA also identified the empathy networks of relevance for us, which was interesting. The only, when we did the Mancover, sympathy was kind of showed support for the cognitive empathy, contributes to the sympathy. The IC, which actually had the cognitive empathy areas, contributes to the sympathy only. So sympathy seems to be something that people do feel, but not so much empathy or pity or compassion. So the next one was on faces. So we generated 24 stimuli. These are the faces which are the average Asian faces that are generated by University of Florida. And what we did was we photoshopped them to kind of look, put some features on it to look Indian. So you had the bindi and change the skin tone color. So we had 24 stimuli. Each one were 4 of a set. And for every one of them, we gave the information saying that these are rape victims. Okay, nothing else, just the face was shown. So we use this word traditional here. That is to say, any face with a bindi and hair tied back is considered traditional. This was as labeled by people. This was after pre-experiment. This was labeled. Non-traditional or quote unquote modern, as it is called in India, is when you leave your hair and you don't show any sign that like bindi or wearing this is typically what we can get from the common society of India. So urban and rural, we just wanted to see if, say, the victim is from a village as against a city. Okay. So when we did this, what we found, the first sample t-test was not interesting, but the second one, most interesting, was the only areas, the areas supporting empathy which survived were for the traditional. That is where the right insula, this insula is very critical for emotional empathy, and only for traditional you had this survived. And of course even for color, but then we had the right and the left insula, but dark versus light, but when we added more analysis, it didn't survive. So traditional faces actually had higher emotional empathy areas, activation in areas supporting emotional empathy survived for the traditional faces as against the non-traditional faces. This was the final takeaway, but again, it is uncorrected. And this also kind of supports the scoring that they gave for traditional faces, which was higher in terms of the empathy scores as compared to the rest of them. So these are the major findings. I'll quickly go through it. I think I'm way behind time. So correlation analysis of the RMA, that is the rape myth acceptance, showed that the one where the doubt is on the victim and she asked for it. These are two which basically places the onus of responsibility on the rape victim, showed high and positive correlation. And the ones where it wasn't really rape, that means it benefits of doubt is on the rapist, and he didn't mean to. There was a distinction between face categories. The distinction between face categories was not very clear, but for it wasn't really rape. The non-traditional face response is dependent on rejection of the myth, while traditional faces show no such correlation. So what is the major takeaway? So the face appearance, when categorized as non-traditional, would mean higher acceptance of the myths. Okay, and both the fMRI study from the narratives and from the behavioral data shows that we have a cultural biases on the concept of what is traditionality, as defined. I don't know by whom, but it is still it exists. Whichever script wrote it. There are some limitations, as all fMRI studies are, something that I think every paper actually talks about it. We had funds only to kind of do scanning of 20 participants, and any long duration like 80 seconds has its own problems with fMRI analysis. So we are now looking at objectification. I'll quickly go through the first experiment. So objectification is basically when you break up somebody into, you perceive somebody to be an object. That means you are shown somebody standing in front of you. You focus on certain parts of the body, which basically reduce that person into being an object and not consider the person as a human as a whole. Okay, so these are standard studies done across the world on objectification. They usually use images with attires which are like either a bikini or low, a slightly more sexualized kind of an attires. But we used a regular pant and shirt because we said we don't even have to go to that extent. We just can use woman with pant and shirt, and that itself can give us the responses we need. So we use the NEVON local and global recognition because it has been shown that local and global recognition, the way the NEVON responses that we have for local and global could also can also reflect on the way we objectify. Okay. So participants were 94, 24 women and 70 men. Most of them were students. And what were the images shown? We just showed images like that. We also used male actor images. These are common images found on the net. We made them black and white so that we don't have color problems. And these were just images like this. These are clothes that all of us wear when we go out. And all they had to do was, this was eye tracking study. So they actually see the image and they are primed with the NAVON. They have to either identify the local letter or the global letter. Okay, the response time was collected. And the data analysis was whether the priming condition was a male or the female actor images, and the recognition test was the local recognition or the global recognition. And the other data that we collected was visit duration, fixation time, and selected AOI. So what you're seeing here is the heat maps that are generated after the eye tracking. Basically, it shows where our scans are. So we took 3 AOIs, that was areas of interest: the face, the chest, and the hips. And we did this for both the images. And what we found was, this is the first set of analysis. My students, Rejji, are still working on it. So the interesting thing we found, which is contrary to the similar studies done across the world, is that face seems to be very important in the Indian culture. So the male face had, of course, more number of time was spent on the male face than the female face. And all of it was statistically significant. And the total time spent on face is greater than the total time spent on the chest and the hips. It is irrespective of whether it was a male image or a female image, and this is interesting. And but the total time spent on a female chest and hips is greater than that was spent on the male. Okay, and most interesting and actually also confirms the initial bias we found in respectability is both men and women sexually objectify women more than men. And now this, you can say intentions are not the same, but visual scanning of the body is similar. So this also relates to what we call a self-objectification that all of us, whether it's a male, female, or other gender, do. All of us do, is self-objectification, and this reflects on the way we scan another person who is in front of us. So the only difference we found as compared to other similar studies across the world, is that the focus on the face is more. And we found, and when we did the behavioral study after that, the face is important for us because it also gives us a lot of, the participants said it also gives them a lot of other feature sets, things like their social status, the color, the beauty, attractiveness is the word used. So that's important for them. So, but if not, the face was the only difference we found from the Western studies to here is the face, the concentration on the face. We've also done this study, which I can't show the results now, it's still being analyzed, where we used two videos. One was a sexually objectified movie song that is there in all of Bollywood, Hollywood, whichever would you take, and so-called neutral kind of a song, which doesn't have so many camera shots or spans that cover or zoom in onto different parts of the female body. And we did the priming study where we used images of women with sari dress and all those things. But yes, this is a continuing work and a lot of things. But the most important thing that I wanted to actually come and talk in this session was, everywhere we go, they ask us, so yes, most of it we know. So what is the interventions you can help us with? I think we need to come up with interventions as a collective. Where do we address it? What do we address? I think those are all, we don't have to actually get into which is the most important thing. Most important thing is obviously in front of us, is social biases. Where do we start? We should start from children. We've heard, we have taken data unstructured from very young children, nothing to do, I mean, we didn't show them any of these just a class kind of an interaction, and they talk about how they take role models, okay, and the stereotypes they have. I think interventions is what this group, this committee, this movement should actually talk about. We need to address this fast. So thank you, and I would like to acknowledge DST-CSRI funding that started in 2019, which is on the objectification and then the alcohol effects. And thanks to NBRC, and special thanks to Nandini. Without her, we could not have been able to do the scanning and all the participants of the study. Thank you.
Thank you, Dr. Vemuri. That was really a very insightful talk on these socially relevant issues, social biases, but at the same time, very difficult problems. Unfortunately, we are kind of really running out of time. But there are a few questions for you in the chat box, if you don't mind answering there. And also, I would just wanted to make a note. I think I misspoke when I was introducing. I said IIT, it's IIIT Hyderabad. So I just wanted to correct that. And
Yes.
Thanks a lot again for your talk.
Thank you. Thank you. Bye-bye. I'll answer the questions.
Okay, thank you. I'm sorry about that. It's just like it pressed for time. Okay, and finally, we move on to our final speaker for this session. And please extend a very warm welcome to Dr. Rejji Kuruvilla. She's a professor at Johns Hopkins University, Baltimore. And she and her team study the development and functions of the sympathetic nervous system using various approaches from mouse genetics to biochemical, imaging, and physiological analysis. Particularly, her work investigates how the connectivity of the sympathetic nervous system is established and refined by the actions of neurotropic signals secreted from target tissue, and in turn, how the sympathetic nerves instruct target tissue development and function. Along with all her tremendous research contributions in the field, she is the organizing chair of the 2021 Gordon Conference on Neurotropic Factor Functions in Development, Plasticity, Regeneration, and Disease, is a member of the Special Emphasis Panel for Endocrinology, Metabolism, Nutrition, and Reproductive Sciences Integrated Review Group for the US National Institute of Health. And for today, she'll be representing the neuronal development field and give a talk on studying the two-way relationship between sympathetic neurons and their target tissues, focusing primarily on pancreatic function and glucose homeostasis. Dr. Rejji, thanks a lot for accepting our invitation to give a talk in this conference, and the floor is all yours.
Share my screen. Okay. So thank you very much to the organizers for inviting me and for putting together a really fantastic series of talks. Given the time difference, I'm sorry I could not attend most of the talks, but I really would like to comment the organizers for highlighting inequities in academia. I think having diversity in academia enriches all of us, brings different perspectives, and is just the right thing to do. Now, this is the first time I've actually woken up at 4 a.m. to give a talk, so please excuse me if there are any missteps, but I'm really delighted to be here, looking forward to meeting with students and postdocs and early stage researchers later on. Can you see my screen?
Yes, absolutely. It's perfect.
Awesome. So my lab, we are interested in how neuron development is shaped by interactions with the target tissues, and to form functional connections during development, neurons often receive specific instructions in the form of soluble cues secreted by their targets. Some of these cues act locally at the nerve terminals to acutely influence local growth events. And in some cases, these signals have to be transported long distance, millimeters or meters away to the cell bodies or the nuclei to activate cytoplasmic and transcriptional signaling necessary for neuron survival, axon growth, and maturation. Now, to date, the family of neurotrophins provides one of the best examples of these target-derived instructive cues. They are small peptide factors, about 13 kilodaltons in size, and they act through receptors belonging to the TRAC receptor tyrosine kinase family. The founding member of this family of neurotrophins is NGF, a nerve growth factor, which will be the focus of my talk today. Other neurotrophins that were later identified include BDNF, that has diverse effects in the brain, neurotrophin 3 and neurotrophin 4. Now, together, this small group of growth factors controls an astonishingly diverse array of effects in the vertebrate nervous system. Some of the effects, such as the regulation of survival and axon growth during development, can be long-term. Other effects, such as the modulation of neurotransmitter release and synaptic plasticity, is on the order of milliseconds. Now, work that we and others have done has shown that during development, sympathetic, just to quick, one example of neurons that rely on these neurotrophic cues are neurons in the sympathetic nervous system. These neurons are responsible for preparing our body for fight or flight in response to stress or danger, but they're also responsible for maintaining body homeostasis during daily activities such as eating a meal, exercise, or just control of body temperature. Now, the neurons the cell bodies are clustered into ganglia that lie in a chain parallel to the spinal cord, and their axons, shown here in red, will reach and innervate almost every organ and tissue in the periphery to control fundamental physiological processes that affect our daily lives. This include pupil dilation, heart rate, sweating, and glucose metabolism. Now, dysfunctional sympathetic nervous system has been linked to several prevalent disorders that are shown here, and typically not what we consider disorders of the nervous system. And what has been of interest to our lab is emerging evidence that early defects acquired during development could contribute to some of this pathology. Now, work that we and others have done has shown that during development, these axons will often travel along blood vessels to reach and innervate their final targets. Shown here is the sympathetic nerves in the entire pancreas of a 3-week-old mouse. The nerves have been labeled with the noradrenergic marker, tyrosine hydroxylase. And you can't see the cell bodies here because the cell bodies are somewhere to the top right. Now, the axon extension and branching of these nerves in these target organs relies on the target-derived neurotrophin, NGF. In fact, the very survival of these neurons relies on NGF. In knockout mice lacking NGF or its TRK receptor, 100% of these neurons are lost within the first 2 weeks after birth. So here you can see a pancreas in the mouse in which the sympathetic fibers have been completely lost. So I have a quick question. Can you see my presenter view, or are you seeing the correct screen?
Yeah, we can see your pointer as well.
Okay, I just wanted to make sure that you are not seeing the presenter view.
No, this is
Okay.
perfect. Yeah, thank you.
So given that this neurotrophin, NGF, is made by the target tissues and not by the neurons themselves, a fundamental cell biological question has been to figure out how it signals in these polarized neurons. And work that we and others have done support a model where endocytosis and trafficking of the TRK receptors are important for NGF's actions. So from work done in my lab and other labs, we know that when the ligand binds to TRK receptors on the axons, this results in the internalization of ligand receptor complexes into vesicles that have been called signaling organelles, so-called because the receptor continues to be active from within these intracellular locations. These ligand receptor complexes are then retrogradely transported along the axon back to the cell bodies where they continue to signal to regulate changes in gene expression. So some of the major questions that are outstanding in the field that we and others are addressing is about the cellular and molecular mechanisms that underlie this long distance signaling and trafficking from the axon back to the cell bodies of the nuclei. Specifically, what are the pathways that are involved in receptor endocytosis and long distance trafficking along the axon? What is the complete proteomic profile of these signaling organelles? There's even some debate in the field as to the exact identity of these organelles, where some groups have defined them as endosomes, other groups have defined them as multivesicular bodies. What is the fate of these signaling organelles when they reach the cell bodies, and how do they communicate with the nucleus to regulate transcriptional events? So these are some of the questions that we and others are interested in addressing in this context. Now, for today's talk, I'm going to actually focus on the role of neurotrophins that have been classically studied as neuronal growth and survival factors, as well as sympathetic nerves, in the development and functioning of a peripheral tissue. Now, of all the organs and tissues innervated by sympathetic axons, we chose to focus on the pancreas. Now, the pancreas, as you may know, serves two major functions. The exocrine part of the pancreas, which makes up most of the pancreatic tissue, secretes digestive enzymes into the gut. The endocrine pancreas, characterized by these clusters, islets of Langerhans, controls blood glucose homeostasis through the release of hormones such as insulin and glucagon. Now, here in a mouse islet, you can see insulin producing beta cells that are located at the core or the center of the islet, and glucagon producing alpha cells, as well as other endocrine cell types at the periphery. Now, several years ago, we noted that the neurotrophin receptors, the TrkA receptors that have been studied in the nervous system, are also expressed in pancreatic beta cells that make insulin. We were not the first to make this discovery, because it has been known for a long time that these neurotrophin receptors are found outside of the nervous system in many non-neuronal tissues. But what these receptors were doing in non-neuronal tissues had remained somewhat of a mystery. So the work that I'm going to describe in the next few slides has been that of a very talented graduate student, Jessica Houts, who's now doing a postdoc at Scripps, Florida. Now, to define the pancreas-specific functions of TrkA receptors, Jessica decided to conditionally delete them in mice using the Cree-Loxp system, and I noted that Poonam had introduced this approach earlier. And what she noticed was a very striking defect in glucose tolerance in adult mice. So typically, after fasting mice overnight, mice will have a blood glucose level of about 100 mg per deciliter. If you inject them with a bolus of glucose, this climbs, the blood glucose levels climbs to about 300 mg per deciliter in the control mice, shown in the black lines, and subsequently returns to baseline over 2 hours. This is because of insulin secretion by the pancreas, as well as glucose uptake by peripheral tissues such as the liver, adipose tissue, etc. But in the mutant animals shown in red, you will notice that the blood glucose excursions after the glucose challenge are much higher. Now, when Jessica looked at the plasma insulin in response to a glucose challenge, the wild type animals shown in the black lines showed a spike in insulin after the glucose challenge, whereas this effect was blunted in the mutants. So together, these results provided the first evidence that TrkA receptors, again classically considered to be functioning in growth and survival of neurons, are required for glucose homeostasis and insulin secretion at the whole animal level. Now, since in these mutant mice, we were deleting TrkA at early embryonic stages, we did not know if these metabolic phenotypes came from early developmental defects, or did this reflect a role for TrkA in mature beta cells. So to distinguish between these possibilities, we used a chemical genetic approach in mice that would allow us to inducibly silence the receptor kinase activity at adult stages. So we used knock in mice, where the TrkA receptors have a modified ATP binding pocket such that they are now vulnerable to a small molecule inhibitor 1-NMPP1. So we confirmed that this drug inhibits TRAC phosphorylation in isolated islets from the knock-in mice, but had no effect on the wild-type islets. And remarkably, injection of the small molecule inhibitor to TRAC-A just 20 minutes prior to the glucose challenge resulted in impaired glucose tolerance, as well as blunted insulin secretion, very similar to the phenotypes that we had seen with the genetic loss of the TRAC-A receptors. So these results suggested that TRAC-A activity is acutely required for insulin secretion in mature beta cells in a manner independent of development. So then the next key question that we asked is, what is the source of the ligand NGF in the pancreas? So previous studies had reported that in cultured beta cells make NGF, which could then act in an autocrine manner to modulate insulin secretion. Surprisingly, when we deleted NGF from all pancreatic cell types, including beta cells, this had no effect on glucose tolerance. And when we looked at insulin secretion in isolated islets, the loss of NGF from pancreatic beta cells had no effect on insulin secretion. So here in the black bars, you can see wild-type islets when respond to elevated glucose by secreting more insulin. And this effect is similar in the mutant islets shown in red. So these results suggested that in contrast to the cultured studies in culture, the pancreatic beta cells are not the relevant source of NGF for insulin secretion. So if not beta cells, which other cell types in the pancreas are producing NGF? So we used a genetic reporter mouse line where LAG-Z reports NGF expression, and we observed robust NGF expression actually in the pancreatic vasculature. So the arrows show beta-gal activity in the large diameter blood vessels that are outside of an islet, outlined in red. And on the right, the arrowheads show NGF expression in the islet microvasculature inside the islets. So using immunostaining, we determined the NGF expressing cell types outside of the islets to be actually the vascular smooth muscle cells defined by the marker smooth muscle actin. And inside the islets, we identified the NGF expressing cell types to be pericytes. They are a vascular smooth muscle-like cell type that had been poorly studied. So we did not see any NGF expression in the endothelial cells. So together, these results suggested that vascular smooth muscle cells, either smooth muscle cells in the large diameter blood vessels, or these pericytes are the source of NGF for insulin secretion. Now, to test this prediction, we once again use the Cree-Lox-P system to remove NGF from vascular smooth muscle cells. And this time, in sharp contrast to removing NGF from pancreatic beta cells, these mice where NGF was removed from vascular smooth muscle cells showed impaired glucose tolerance, as well as reduced insulin secretion to a glucose challenge, similar to the effects that we had seen with either the loss of TRAC-A or the silencing of TRAC-A activity. So to summarize what I have described so far, these results suggest a neurotrophin, NGF, as a non-canonical role for a neurotrophin as a vascular-derived paracrine factor that promotes insulin secretion. So our results suggest that NGF secreted by vascular pericytes acts on TRAC-A receptors in neighboring beta cells to promote insulin granule exocytosis. So in data that I'm not presenting, we found that this effect of NGF is likely in the secretion of insulin and not in the biosynthesis of insulin. So then a key question that we asked is, how does this NGF pathway intersect with glucose? From all that we've learned in textbooks, glucose is the major trigger for secreting insulin from beta cells. So is NGF TrkA signaling active at all times and is co-opted by glucose to enhance insulin secretion? Or is this pathway actually regulated by high glucose? And our results suggest that the latter scenario might be the case. So here we found that high glucose concentrations, 16.7 millimolar glucose compared to the low glucose, promotes secretion of NGF from vascular smooth muscle cells, but more importantly, results in a striking increase in TrkA phosphorylation in isolated islets. Now, this glucose treatment was only for about 15 minutes. So again, we think that this effect is at the level of NGF secretion and not at the level of NGF biosynthesis. So together, these results actually suggest a feed-forward mechanism where high glucose recruits the NGF signaling pathway to augment or enhance insulin secretion. And inside the beta cells, we found that some of the same cellular mechanisms that are used by neurotrophins to promote survival and growth signaling in neurons are also used to promote insulin secretion from pancreatic beta cells. So I'm going to summarize our results to say that we found that NGF binding to TrkA receptors on beta cells promotes receptor endocytosis, just as it does in neurons. Receptors signal from within these endosomes to remodel a dense actin meshwork that acts as a barrier to insulin granule exocytosis and facilitates insulin granule release. So these results suggest that neurotrophins use conserved mechanisms to elicit different functional outcomes in different cell types: survival and growth signaling neurons versus insulin secretion in pancreatic beta cells. Now, you may ask, if glucose is a major trigger for secreting insulin from beta cells, why do you need additional signals such as NGF? And it's been known for a while that there is multifactorial control of insulin secretion from beta cells. This is in part because the glucose-triggered pathway for insulin secretion is inefficient. And secondly, there is a need to precisely coordinate insulin secretion from pancreatic beta cells with the functional states of neighboring tissues, such as the nerves and the vasculature or other tissues. We know that some of these insulin modulatory signals come from afar, carried through the bloodstream. So it's been known for a while that intestinal hormones, as well as nutrients derived from a meal, can modulate glucose-stimulated insulin secretion. We also know that there are local factors within the islet microenvironment, one key example being neurotransmitters that are released from the nerve terminals innervating the islets that can also modulate glucose-triggered insulin secretion. And we now add neurotrophins as another paracrine factor within the islet microenvironment that regulates the ability of glucose to promote insulin secretion. And we think that maybe this reflects a need to coordinate insulin secretion with local changes in the islet vasculature. We are very interested actually in the islet pericytes that have been very poorly studied, how they respond to high glucose, and what are the mechanisms by which they release these neurotrophins. Now, in addition to this direct effect of NGF on the beta cells, NGF can also indirectly affect islet development and function by recruiting sympathetic nerves. So shown here in this image, pancreatic islets are richly innervated by NGF responsive sympathetic nerves. Shown in white are sympathetic nerves that actually track along the blood vessels, shown in red, to reach and wrap around an islet that has been stained for insulin in cyan. Now, given this close association, we asked if the nerves might contribute to aspects of islet development or function. Now, in adult islets, it's well known that sympathetic activity acutely suppresses insulin secretion and promotes glucagon secretion. And the net effect is to increase the circulating glucose in your bloodstream to enable you to run away in a dangerous situation or also during a stress response. But these sympathetic fibers reach the pancreas during embryonic stages, and in mice as early as embryonic day 13.5. And this is a time when these pancreatic progenitor cells are emerging from this ductal epithelium. They are undergoing periods of rapid growth, proliferation, differentiation, and maturation. And the contribution of the nerves to these aspects of islet development had not been addressed. And this was a question that was addressed by a previous graduate student, Phil Borden. So Phil decided to take a genetic approach to ablate sympathetic nerves during development by conditionally removing the TrkA receptor from sympathetic neurons. And as I introduced before, this results in the loss of 100% of the neurons within the first 2 weeks after birth, and a complete loss of innervation to several target tissues, including the pancreas as shown on the right. Now, what Phil noticed in these denervated or sympathectomized mice, as I will call them from now, was a profound impairment of the islet architecture at neonatal stages. So on the left is, you see the characteristic arrangement of a mouse islet with the insulin-producing beta cells at the core, and this is thought to facilitate cell-cell contacts between the beta cells that promotes optimal insulin secretion surrounded by these glucagon producing alpha cells. In the mutants, the islets appeared as these streak-like aggregates with an intermingling of the alpha and beta cells. And in data that I'm not providing, we found that significant disruption in the cell-cell contacts between the beta cells. Now, when Phil looked at metabolic parameters in these sympathectomized mice at adult stages, he found that these mice showed reduced insulin secretion in response to a glucose challenge, as shown in the red lines. And they were also glucose intolerant, as shown again in the red line. So these results provided the first evidence that sympathetic nerves during development is essential for establishing islet structure and for later function. So since then, a major question that's been driving our lab is to identify the nerve-derived factors that are responsible for islet organization and function. Now, the sympathetic nerves that innervate the pancreas secrete the neurotransmitter norepinephrine, which acts through adrenergic receptors that are GPCRs located on the islet cell types. Now, in data that I'm not showing, we found that in cultural studies, nerves could promote the migration and clustering of the beta cells, which could be prevented by blockers of these adrenergic receptors. So these results hinted that sympathetic neurotransmission or activity during development could be involved in aspects of islet formation and function. Now, to define the role of norepinephrine as well as sympathetic neurotransmission, we again used a genetic approach to express the tetanus toxin light chain subunit in sympathetic neurons in mice. Now, tetanus toxin cleaves the synaptic vesicle protein VAM2, which prevents synaptic vesicle fusion and release and inhibits norepinephrine release from these neurons. Now, in these silenced mice, as I will call them from now, so briefly, this is the work of a postdoc in the lab, Eugene Lin. Now, in these silenced mice, we found that silencing of sympathetic activity during development did not compromise the ability of the nerves to reach and innervate their targets. So here you can see sympathetic fibers in white in both the control and in the mutant pancreas. But what you will notice immediately is a distortion of islet morphology, similar to the phenotypes that we had seen when we genetically ablated the nerves. Further on, when we looked at both insulin and glucagon immunostaining, we found again the mislocalization of insulin and glucagon positive cells, as we had seen with the ablation of the sympathetic nerves. So these results suggest that loss of sympathetic nerves or silencing of activity during development results in similar disruptions in islet morphology. Well, what about function in these silenced animals? We found that inducibly silencing sympathetic neurotransmission during development indeed resulted in glucose intolerance in adult mice at later stages, but these effects were very mild compared to the effects of removing the nerves, as shown on the right. So you can see that the effects of the glucose intolerance in the sympathectomized mice is much more severe than that in the silenced mice. So these results suggest that sympathetic neurotransmission contributes in part to the developmental effects of nerves on islet morphology and function, but is not the whole story. So this is ongoing work in my lab, together with our previous work that showed that these neurons are supported by target-derived neurotrophic factors such as NGF. Our new work shows that these neurons in turn release factors, including norepinephrine, that support the development and function of the target pancreatic endocrine cell types. We are very interested right now in identifying other nerve-derived factors in addition to norepinephrine that influence aspects of islet maturation and development. We are using electron microscopy and 3D imaging to define the nature of the contacts between the nerves as well as the pancreatic endocrine cell types. And we're also performing RNA sequencing analysis from islets where the sympathetic nerves have been removed or sympathetic activity has been silenced to see how this affects the transcriptional signatures of the islet cell types. So I've given credit to people who did the work along the way. This is my lab before the COVID and time when we could meet in person. And this is the building, biology building at Johns Hopkins University on our campus. I would like to thank the funding sources, and I will stop here and ask questions and take any questions, sorry. Thank you very much for your attention.
Thank you, Dr. Rejji. That was a wonderful talk on the regulation of the islet functions and by the NGF and the sympathetic nervous system. Unfortunately, we are running out of time, and we'll be going into the breakout rooms right after. So there are quite a few questions for you, as you can see in the chat box, but we can also discuss those questions. I mean, you can answer in the chat or also in the breakout rooms. So sorry about that, but we really want to thank all the speakers for a wonderful series of talks today, and all the audience for being so engaging throughout the entire session. Just wanted to remind a couple of things. So we have the breakout session would happen right after this. So you would have already received your specific the Zoom links for the breakout session, and please use them to head to your respective breakout rooms. And we will be returning to our public forum again after a short break at 4:30 for the ALBA network session. And this is for the bias in Indian STEM. And so the members of the audience, please use the separate Zoom link for this event, which we have emailed to you to re-enter the conference. And also, we have a special message for the Ungender workshop for the gender awareness and bias sensitization, which is happening for April 12th. And in order to get the most benefit out of this session, we encourage interested audience members to fill out a short and anonymized Google form with the questions related to their attitudes and suppositions regarding biases encountered in their daily lives. And the link for that is available in the chat. So hopefully you can access that. Okay. So for the ALBA session, again, just to remind you, it would be happening at 4:30 p.m. IST. And given the spirit of that session and to foster a spirit of inclusivity and openness throughout this conference, we highly request all our audience members to think about adding their preferred pronouns to their Zoom names in solidarity with people of underrepresented genders. You can also do this by adding preferred pronouns in brackets after your name in the Zoom profile before logging into the ALBA session. And members of the audience, please use the separate Zoom webinar link for this event that we have emailed to you to re-enter the conference. So hope to see you all in half an hour at the ALBA Network session on bias in Indian STEM. And for now, we'll go right into the breakout rooms. Thank you, everyone.